Right now, as you settle into this moment of quiet learning, 3 pounds of neural tissue nestled within your skull are orchestrating an intricate symphony of life. Your brain is simultaneously monitoring your heartbeat, processing these words, storing memories of your day, and perhaps already beginning the gentle transition towards sleep. This remarkable organ, no larger than two clenched fists, contains roughly 86 billion neurons connected by trillions of synapses, creating networks more complex than any supercomputer ever built. Tonight, we embark on a journey through these neural landscapes, exploring how this extraordinary command center transforms electrical impulses into thoughts, emotions, dreams, and the very essence of what makes you uniquely human. If you could shrink yourself down and journey through the neural pathways of your own brain, you would find yourself exploring a landscape more varied and intricate than any earthly terrain. This 3 pound universe nestled within your skull contains distinct regions, each with its own geography, purpose, and personality. Understanding this landscape begins with recognizing the brain's major divisions, each as essential to your existence as the organs beating in your chest or the air flowing through your lungs. The human brain, when viewed from above, resembles a wrinkled walnut split down the middle. This largest portion, the cerebrum, dominates the cranial cavity like a vast mountain range of folded tissue. Its surface, the cerebral cortex, contains the neural real estate responsible for everything we consider distinctly human: language, reasoning, creativity, and conscious thought. Beneath this cortical landscape lie deeper structures that govern emotion, memory, and the countless automatic processes that keep you alive moment by moment. The cerebrum divides naturally into two hemispheres, left and right, connected by a thick bundle of nerve fibers called the corpus callosum. This bridge allows the two sides of your brain to communicate constantly, sharing information and coordinating responses. Each hemisphere controls the opposite side of the body, a curious arrangement that means your left brain directs your right hand as it turns these pages, while your right brain processes the spatial relationships of the words on the page. Each cerebral hemisphere subdivides further into four distinct lobes, named for the skull bones that protect them. The frontal lobe, largest of all, extends from your forehead back to roughly the crown of your head. This region houses your personality, decision-making abilities, and motor control. The parietal lobe, situated behind the frontal lobe, integrates sensory information and maintains your sense of body position in space. The temporal lobe, nestled beneath the parietal region near your temples, processes auditory information and plays crucial roles in memory formation. Finally, the occipital lobe, occupying the back of your head, transforms electrical signals into the rich visual world you perceive. Tucked beneath the cerebrum's posterior edge lies the cerebellum, Latin for little brain. Though smaller than the cerebrum, this structure packs an extraordinary density of neurons into its tightly folded surface. The cerebellum resembles a miniature brain in its own right, with its own hemispheres and cortex. This region serves as your body's movement coordinator, fine-tuning motor commands from the cerebrum and maintaining balance and posture. Recent research reveals the cerebellum also contributes to cognitive functions, including language processing and emotional regulation. Connecting the cerebrum and cerebellum to the rest of your body is the brainstem, a structure that might appear unremarkable but performs perhaps the most vital functions of all. This narrow column of neural tissue extends from the base of the brain down to the spinal cord, serving as the critical junction between mind and body. The brainstem consists of three main sections: the midbrain at the top, the pons in the middle, and the medulla oblongata at the bottom. The midbrain, though small, houses important centers for visual and auditory reflexes, eye movement control, and the production of dopamine, a neurotransmitter crucial for movement and motivation. The pons, meaning bridge in Latin, serves as a relay station for information traveling between the cerebrum and cerebellum, while also containing centers that regulate sleep, arousal, and facial sensation. The medulla oblongata controls your most fundamental life functions: breathing, heart rate, and blood pressure. Even now, as you read these words, your medulla is quietly adjusting your breathing rhythm and monitoring your cardiovascular system. Deep within the cerebrum, hidden from view, lies a collection of structures collectively known as the diencephalon. This region includes the thalamus, a pair of egg-shaped structures that serve as relay stations for sensory information traveling to the cortex. Nearly all sensory input, except smell, passes through the thalamus before reaching conscious awareness. Adjacent to the thalamus sits the hypothalamus, a structure no larger than an almond, but wielding enormous influence over your body's internal environment. The hypothalamus regulates hunger, thirst, body temperature, and hormonal cycles, maintaining the delicate balance necessary for survival. This intricate neural landscape requires constant protection and nourishment to function properly. Three layers of protective membranes, collectively called the meninges, envelope the brain like a biological security system. The outermost layer, the dura mater, is a tough, fibrous membrane that adheres to the inner surface of the skull. Beneath this lies the delicate arachnoid mater, named for its spider web-like appearance. The innermost layer, the pia mater, clings closely to the brain's surface, following every fold and crevice. Between the arachnoid and pia mater flows cerebrospinal fluid, a clear, colorless liquid that serves multiple vital functions. This fluid cushions the brain against impact, provides buoyancy that reduces the organ's effective weight, and delivers nutrients while removing waste products. Your brain produces approximately 500 milliliters of cerebrospinal fluid daily, continuously replacing this protective bath. The fluid circulates through a series of interconnected chambers called ventricles within the brain before flowing around the brain's surface and eventually being reabsorbed into the bloodstream. The brain's blood supply represents one of the body's most carefully regulated systems. Despite comprising only about 2% of your body weight, your brain consumes roughly 20% of your total energy supply. This voracious appetite for fuel requires a robust vascular network capable of delivering glucose and oxygen while removing metabolic waste. Four major arteries supply the brain: two carotid arteries running up the front of the neck, and two vertebral arteries ascending through the spine. These vessels interconnect at the brain's base, forming the circle of Willis, a remarkable redundant system that ensures blood flow even if one artery becomes blocked. The brain maintains strict control over what substances can cross from the bloodstream into neural tissue through a selective barrier called the blood-brain barrier. This biological security checkpoint protects the brain from toxins and pathogens while allowing essential nutrients to pass through. Specialized cells lining the brain's blood vessels form tight junctions that create this selective barrier, ensuring your neural tissue remains in a carefully controlled environment. Understanding this anatomical foundation reveals how your brain's structure reflects its function. The cortical folding increases surface area dramatically, packing more neurons into the limited space of your skull. The layered organization allows for complex processing networks, while the deep structures maintain the basic functions necessary for survival. The protective coverings and specialized blood supply ensure this delicate tissue can operate continuously throughout your lifetime. Each region of this neural landscape contributes to the unified experience of consciousness, yet no single area works in isolation. The frontal lobe's executive decisions rely on sensory input from the parietal lobe, emotional context from deeper limbic structures, and motor coordination from the cerebellum. This integration occurs through vast networks of connecting fibers that link distant brain regions in sophisticated communication webs. As you prepare to journey deeper into this neural territory, consider the remarkable fact that every thought, emotion, and sensation you experience emerges from the coordinated activity of these anatomical structures. The words you're reading activate your occipital lobe's visual cortex, while your frontal lobe processes their meaning, and your temporal lobe accesses memories that give them personal significance. Meanwhile, your brainstem continues its tireless work, maintaining the breathing and circulation that keep every neuron functioning. This landscape of the mind, with its mountains of cortex, valleys of white matter, and rivers of cerebrospinal fluid, provides the stage upon which the drama of consciousness unfolds. Each anatomical structure represents millions of years of evolutionary refinement, shaped by the demands of survival and the emergence of human intelligence. As we move forward in our exploration, we'll descend from this bird's eye view of brain geography to examine the cellular inhabitants that bring this neural landscape to life. Within the vast neural landscape we've just explored, a microscopic universe of extraordinary complexity unfolds. If we could shrink ourselves to the size of molecules and wander through the tissue of your brain, we would discover that consciousness itself emerges from the intricate dance of billions of specialized cells, each no larger than a few hundredths of a millimeter across. These cellular architects of thought work together in networks so sophisticated that they give rise to everything from your memories of childhood to your dreams of the future. At the heart of this cellular symphony are neurons, the brain's primary information processors. Each neuron is a living electrical cable capable of receiving, integrating, and transmitting signals across vast networks that span your entire nervous system. Unlike other cells in your body, neurons have evolved a unique architecture specifically designed for communication. Their elongated, branching structure allows them to reach across surprising distances, forming connections that can stretch from your spinal cord to your toes or create intricate local networks within a single brain region. A typical neuron consists of three main components, each specialized for a different aspect of cellular communication. The cell body, or soma, houses the nucleus and most of the cell's metabolic machinery. This central hub contains the same organelles found in other cells, mitochondria for energy production, ribosomes for protein synthesis, and an endoplasmic reticulum for cellular manufacturing. However, neurons pack these structures more densely than most cells, reflecting their high energy demands and constant need for new proteins to maintain their extensive projections. Extending from the cell body like branches from a tree are the dendrites, whose very name derives from the Greek word for tree-like. These delicate projections serve as the neuron's receiving antennas, studded with specialized receptors that detect chemical signals from other neurons. The dendritic tree of a single neuron can be remarkably elaborate, with some cells sporting thousands of branches that create a three-dimensional canopy capable of receiving input from hundreds or even thousands of other neurons simultaneously. The third component, the axon, represents the neuron's transmission line. This single projection can extend from a few micrometers to over a meter in length, carrying electrical signals from the cell body to distant targets. The axon maintains a constant diameter throughout its length and typically branches only at its terminal end, where it forms connections with other neurons or target organs. Many axons are wrapped in a white fatty substance called myelin, which acts like insulation on an electrical wire, dramatically increasing the speed at which signals travel. The diversity of neuronal types in your brain reflects the varied functions they perform. Motor neurons, with their long axons extending from the brain to muscles throughout your body, specialize in controlling movement. Their cell bodies cluster in specific brain regions, while their axons bundle together to form the thick nerve trunks that emerge from your brain and spinal cord. Sensory neurons take the opposite approach, carrying information from receptors in your skin, organs, and sensory organs back to the central nervous system for processing. Within the brain itself, interneurons form the vast majority of all neurons. These cells, whose axons remain within the central nervous system, create the complex circuits responsible for processing, storing, and integrating information. Some interneurons have short axons that connect only to nearby neurons, forming local processing circuits. Others project across vast distances within the brain, linking regions separated by several centimeters and allowing for coordinated activity across the entire organ. The communication between neurons occurs at specialized junctions called synapses, structures so small that thousands could fit on the head of a pin, yet so important that they represent the fundamental units of information processing in your brain. When an electrical signal reaches the end of an axon, it triggers the release of chemical messengers called neurotransmitters into the tiny gap between neurons. These molecules drift across the synaptic space and bind to receptors on the receiving neuron, potentially triggering a new electrical signal that continues the chain of communication. This synaptic transmission represents one of biology's most elegant solutions to the problem of cellular communication. Unlike simple electrical conduction, synaptic transmission allows for modulation, amplification, and integration of signals. A single neuron might receive synaptic input from thousands of other neurons, each contributing either excitatory signals that encourage the cell to fire or inhibitory signals that suppress activity. The neuron integrates all these inputs, firing an action potential only when the combined excitatory input exceeds the inhibitory input by a sufficient margin. The strength of synaptic connections can change over time, a property called synaptic plasticity that underlies learning and memory. When synapses are used repeatedly, they often become stronger and more efficient at transmitting signals. This strengthening can involve changes in the amount of neurotransmitter released, modifications to receptor sensitivity, or even the growth of new synaptic connections. Conversely, synapses that are rarely used may weaken or disappear entirely, allowing the brain to refine its circuits based on experience. While neurons rightfully claim the spotlight as the brain's information processors, they could not function without the support of glial cells, whose name derives from the Greek word for glue. Once thought to serve merely as structural support for neurons, we now understand that glial cells perform essential functions that make complex brain activity possible. In fact, glial cells outnumber neurons in the human brain, comprising roughly half of all brain cells and occupying about half the brain's volume. Astrocytes, the most abundant type of glial cell, extend delicate processes that wrap around synapses and blood vessels, creating intimate associations with both neurons and the brain's vascular system. These star-shaped cells maintain the chemical environment around neurons, regulating the concentration of ions and neurotransmitters in the extracellular space. When neurons fire rapidly, astrocytes absorb excess potassium ions that could otherwise disrupt neural function. They also uptake neurotransmitters from synapses, terminating their action and preparing the synapse for the next signal. Perhaps most remarkably, astrocytes form a critical component of the blood-brain barrier, extending specialized processes called end feet that completely surround brain blood vessels. These end feet help regulate what substances can cross from the bloodstream into brain tissue, maintaining the carefully controlled environment that neurons require. Astrocytes also respond to neural activity by triggering changes in blood flow, ensuring that active brain regions receive adequate glucose and oxygen to meet their metabolic demands. Oligodendrocytes represent another crucial type of glial cell, responsible for producing the myelin sheaths that insulate axons in the central nervous system. A single oligodendrocyte can extend processes to myelinate segments of up to 50 different axons, wrapping each axon in multiple layers of lipid-rich membrane. This myelination dramatically increases the speed of electrical conduction, allowing signals to travel at up to 120 meters per second compared to just 1 meter per second in unmyelinated axons. The importance of oligodendrocytes becomes apparent in diseases like multiple sclerosis, where their destruction leads to severe neurological deficits. Microglia, the brain's resident immune cells, patrol the neural environment with remarkable vigilance. These small, highly mobile cells constantly survey their territory, extending and retracting delicate processes that probe the surrounding tissue. When they detect signs of infection, injury, or cellular debris, microglia rapidly transform from their resting surveillance state into activated immune defenders. They can migrate towards sites of damage, engulf pathogens or dead cells, and release signaling molecules that coordinate the brain's immune response. The organization of neurons and glial cells into functional networks represents one of biology's most remarkable achievements. Individual neurons, despite their sophistication, can accomplish relatively little in isolation. However, when connected into circuits containing hundreds, thousands, or millions of cells, they give rise to emergent properties that transcend the capabilities of any single component. These networks can store memories, process sensory information, generate motor commands, and create the subjective experience of consciousness. The simplest neural circuits consist of just a few neurons connected in series or parallel. A reflex arc, for instance, might involve only 3 neurons: a sensory neuron that detects a stimulus, an interneuron that processes the information, and a motor neuron that generates a response. Yet even these simple circuits can exhibit complex behaviors, including adaptation, sensitization, and learning. More complex networks emerge when many simple circuits interconnect, creating hierarchical structures that can process increasingly sophisticated information. In the visual system, for example, individual neurons in the retina respond to simple features like spots of light. These cells connect to neurons in the visual cortex that detect slightly more complex features like oriented lines or edges. These cortical neurons, in turn, connect to cells that respond to even more complex patterns, eventually leading to neurons that can recognize specific objects, faces, or scenes. The brain achieves its remarkable computational power through the parallel operation of countless such networks. While a digital computer processes information sequentially, executing one instruction at a time, your brain processes information simultaneously across millions of parallel pathways. This parallel processing allows the brain to perform feats that still challenge our most powerful computers, such as recognizing a familiar face in a crowd or understanding speech in a noisy environment. The temporal dynamics of neural networks add another layer of complexity to brain function. Networks can exhibit rhythmic oscillations, with groups of neurons firing in synchrony at specific frequencies. These oscillations appear to play important roles in coordinating activity across different brain regions and in organizing information processing. For instance, theta rhythm oscillations in the hippocampus seem to be crucial for memory formation, while gamma rhythm oscillations may help bind together different features of sensory experience into unified perceptions. Recent advances in neuroscience have revealed that the brain's networks exhibit small world properties, combining local clustering with long range connections in a way that optimizes both efficient local processing and rapid global communication. This architecture allows information to spread quickly across the entire brain while maintaining specialized local circuits for detailed processing. The result is a network that can simultaneously maintain distinct, specialized functions while enabling the global integration necessary for unified consciousness. The development of these sophisticated neural networks requires precise molecular mechanisms that guide growing axons to their proper targets and establish appropriate synaptic connections. During brain development, neurons extend growth cones that navigate through the developing tissue using chemical cues, much like following a molecular roadmap. These growth cones can travel remarkable distances, sometimes across the entire developing brain, to find their specific targets among millions of potential connection sites. As we prepare to explore how these neural networks organize into the functional regions that give rise to perception, movement, and thought, it's worth pausing to appreciate the extraordinary nature of the cellular architecture we've just examined. Every moment of your conscious experience, from the simplest sensation to the most complex emotion, emerges from the coordinated activity of these microscopic cellular networks. The words you're reading now activate specific patterns of neural firing that ripple through networks spanning multiple brain regions, creating the subjective experience of understanding and the formation of new memories that may persist for a lifetime. This cellular foundation provides the substrate upon which all higher brain functions operate. As we journey deeper into the brain's organizational principles, we'll see how these fundamental neural building blocks combine into increasingly sophisticated structures, each layer of organization adding new capabilities while building upon the cellular mechanisms we've explored here. As we ascend from the microscopic realm of individual neurons and their networks, we arrive at one of the most remarkable structures in the known universe, the cerebral cortex. This thin sheet of neural tissue, no thicker than a few millimeters, contains the neural circuitry responsible for nearly everything that makes human consciousness unique. Spread across its folded surface are the networks that generate language, creativity, abstract reasoning, and self-awareness. Yet despite its profound importance, the cortex achieves its extraordinary capabilities through an elegant organizational principle that repeats across its entire surface. The cerebral cortex represents evolution's solution to a fundamental engineering challenge, how to pack the maximum amount of neural processing power into the limited space of the skull. Rather than expanding outward like a balloon, which would require an impossibly large head, the cortex folds in on itself, creating the characteristic wrinkles and grooves that give the brain its distinctive appearance. These folds, called gyri and sulci, increase the surface area dramatically. If you could unfold and flatten the human cortex, it would cover an area roughly equivalent to a large newspaper, approximately 2,500 square centimeters, packed into a space smaller than a football. This folded surface maintains a remarkably consistent architecture across its entire expanse. Whether examining tissue from the visual cortex at the back of the brain or the motor cortex near the top, you would discover the same fundamental six-layered organization. This laminar structure represents one of neuroscience's most important discoveries, revealing that the cortex uses a standardized processing module that can be adapted for different functions depending on its connections and inputs. The six cortical layers, numbered from the surface downward, each serve distinct functions in cortical processing. Layer 1, the molecular layer, contains relatively few cell bodies but is rich in the dendritic branches and axons that connect different cortical areas. This superficial layer serves as a highway for long-range communication, allowing distant cortical regions to influence each other's activity. Layer 2 and 3, the external granular and external pyramidal layers, contain smaller neurons that primarily connect to other cortical areas. These layers form the cortex's association networks, linking related processing regions and enabling the integration of information across different sensory modalities. Layer 4, the internal granular layer, serves as the cortex's primary receiving station for incoming sensory information from the thalamus. This layer is particularly well developed in sensory areas, where it contains dense clusters of small neurons, called granule cells, that process the initial cortical representation of sensory input. In the visual cortex, layer IV neurons first extract basic features like edges and orientations from visual input, beginning the process of constructing our rich visual perception from raw sensory data. Layers V and VI, the internal pyramidal and multiform layers, house the cortex's output neurons. Layer V contains large pyramidal neurons whose axons project to subcortical structures, including the brainstem and spinal cord, carrying motor commands and other executive signals. Layer VI neurons primarily project back to the thalamus, creating feedback loops that allow the cortex to modulate its own sensory input. This feedback represents a sophisticated control mechanism, enabling the brain to focus attention on relevant stimuli while filtering out distractions. Within this standardized six-layer framework, different cortical areas develop specialized functions through their unique patterns of connections. Primary sensory areas receive direct input from sensory organs and create the first cortical representations of sight, sound, touch, taste, and smell. These areas maintain precise topographic maps, meaning that adjacent locations in the sensory organ correspond to adjacent locations in the cortex. In the primary visual cortex, for instance, neighboring neurons respond to neighboring points in visual space, creating a detailed map of the visual field across the cortical surface. The primary somatosensory cortex, located in the postcentral gyrus of the parietal lobe, contains a complete map of the body's surface. This map, often illustrated as the sensory homunculus, reveals the disproportionate cortical space devoted to the most sensitive body parts. Your lips and fingertips, rich in touch receptors, claim vast territories of cortical real estate, while your back and arms, less sensitive to fine touch, occupy much smaller cortical regions. This allocation of cortical space reflects the functional importance of different body parts for manipulation and interaction with the environment. Similarly, the primary motor cortex in the precentral gyrus maintains a motor map of the body, with neurons controlling specific muscle groups arranged in an orderly fashion across the cortical surface. The motor homunculus reveals the same disproportionate representation seen in the sensory map, with enormous cortical territories devoted to controlling the intricate movements of the hands, face, and speech apparatus. The precision required for activities like playing a musical instrument or forming speech sounds demands this expanded cortical representation. Primary auditory cortex, nestled within the superior temporal gyrus, organizes sound information according to frequency, creating a tonotopic map where neurons responding to similar frequencies cluster together. Low frequency sounds activate neurons at one end of this map, while high frequency sounds activate neurons at the opposite end, with a continuous progression of frequency preferences across the cortical surface. This organization allows the auditory cortex to decompose complex sounds into their component frequencies, the first step in recognizing speech, music, or environmental sounds. Beyond these primary sensory and motor areas lie the association cortices, regions that integrate information from multiple sources to create more complex representations and behaviors. These areas occupy the majority of the human cortex and show the greatest expansion compared to other species, reflecting their importance in human cognitive abilities. Secondary sensory areas adjacent to primary sensory cortex begin the process of feature integration, combining basic sensory features into more complex patterns and objects. The superior parietal cortex integrates visual and somatosensory information to create spatial maps and guide reaching movements. When you reach for a coffee cup, neurons in this region combine visual information about the cup's location with proprioceptive information about your arm's position, computing the motor commands necessary to guide your hand to its target. Damage to this region can result in spatial neglect, where patients lose awareness of one side of space despite having intact vision. The temporal association cortex specializes in object recognition and memory formation. As visual information flows from primary visual cortex through a series of processing stages in the temporal lobe, neurons develop increasingly complex response properties. Early stages respond to simple features like edges and colors, while later stages respond to complete objects, faces, or even specific individuals. The end stages of this processing hierarchy contain neurons that respond selectively to highly specific stimuli, such as a particular person's face viewed from a specific angle. The frontal association cortex, occupying the anterior portions of the frontal lobe, houses the brain's executive functions. These regions plan complex behaviors, make decisions, and control attention. The prefrontal cortex, in particular, maintains working memory, allowing you to hold information in mind while manipulating it mentally. When you calculate a tip or follow directions to a new location, your prefrontal cortex coordinates the necessary mental operations while maintaining relevant information in an active state. Language areas represent some of the most specialized regions of human cortex, found primarily in the left hemisphere in most individuals. Broca's area, located in the frontal lobe, specializes in speech production and grammatical processing, Damage to this region produces a characteristic pattern of halting, effortful speech with preserved comprehension. Wernicke's area in the temporal lobe processes language comprehension and meaning. Damage here results in fluent but meaningless speech, with patients producing grammatically correct sentences that convey no coherent meaning. Modern neuroscience has revealed that language processing involves extensive networks spanning multiple cortical areas rather than being localized to just these classical language regions. Reading activates visual areas for letter recognition, temporal areas for word meaning, frontal areas for grammatical processing, and motor areas for subvocal articulation. This distributed processing allows the remarkable flexibility of human language, enabling us to communicate abstract concepts, create poetry, and transmit complex ideas across generations. The cortical maps we've described are not static structures fixed at birth, but dynamic representations that can reorganize throughout life. This cortical plasticity allows the brain to adapt to new experiences, recover from injury, and optimize its organization based on use. Musicians who begin training early in life show expanded cortical representations of their fingertips, while London taxi drivers develop enlarged posterior hippocampal regions specialized for spatial navigation. Following brain injury, intact cortical areas can sometimes assume functions previously performed by damaged regions. Stroke patients who lose speech production abilities may recover through the activation of homologous areas in the right hemisphere. Children show even greater plasticity, with the capacity for dramatic cortical reorganization that allows recovery from injuries that would cause permanent deficits in adults. The cortical surface also exhibits a hierarchical organization, with information flowing from primary areas through successive levels of association cortex. This hierarchical processing allows the transformation of simple sensory inputs into complex perceptions and behaviors. In the visual system, the hierarchy progresses from detecting simple features like edges and orientations in primary visual cortex to recognizing objects and scenes in higher order temporal areas to linking visual information with memory and meaning in the most anterior temporal regions. Recent advances in brain imaging have revealed that cortical areas organize into large-scale networks that span multiple lobes and coordinate their activity during specific tasks. The default mode network, active during rest and introspection, includes regions in the frontal, parietal, and temporal lobes that seem to support self-referential thinking and mental time travel. The attention networks include frontal and parietal areas that coordinate to focus consciousness on relevant stimuli while ignoring distractions. These cortical networks operate through rhythmic oscillations that synchronize neural activity across distant brain regions. When different cortical areas need to communicate, they often synchronize their firing patterns at specific frequencies, creating temporary communication channels that allow information to flow efficiently between regions. Gamma oscillations, occurring at 30 to 100 hertzes, seem particularly important for binding together different features of perception into unified conscious experiences. The folded architecture of the cortex also creates natural divisions between functional areas. The major sulci, or grooves, often serve as boundaries between regions with different functions. The central sulcus separates somatosensory cortex from motor cortex, while the lateral sulcus separates temporal cortex from frontal and parietal areas. These anatomical landmarks provide a rough guide to functional organization, though the boundaries between functional areas are often more gradual than the sharp anatomical divisions might suggest. Understanding cortical organization has profound implications for treating neurological and psychiatric disorders. Neurosurgeons use cortical maps to plan operations that remove diseased tissue while preserving critical functions. During brain surgery, patients may remain awake while surgeons stimulate different cortical areas to map language and motor functions, ensuring that vital capabilities are not inadvertently damaged. As we prepare to explore the deeper structures that support and modulate cortical function, it's important to recognize that the cortex, despite its prominence in human cognition, does not operate in isolation. The thin cortical sheet we've examined depends critically on subcortical structures for emotional content, motivational drive, and the basic arousal necessary for consciousness itself. These deeper brain regions, evolutionarily older than the cortex, provide the foundation upon which cortical sophistication builds, creating the integrated neural systems that give rise to the full spectrum of human mental life. Beneath the sophisticated neural networks of the cerebral cortex lies a collection of ancient brain structures that form the limbic system, regions that have been shaping behavior and experience for hundreds of millions of years. These structures, whose evolutionary origins predate the emergence of complex cortical functions, continue to exert profound influence over modern human behavior. They color every experience with emotional significance, forge the memories that define personal identity, and drive the motivations that propel us through life. Understanding these deep brain regions reveals how our most primitive neural circuits continue to guide our most sophisticated thoughts and decisions. The term limbic derives from the Latin word for border, reflecting these structures' position at the interface between the cortex above and the brainstem below. This anatomical location mirrors their functional role as intermediaries between rational thought and instinctual response, between conscious planning and unconscious drive. The limbic system encompasses several interconnected structures, each contributing specialized capabilities to the emotional and memory systems that make human experience so rich and complex. At the heart of the limbic system lies the hippocampus, a curved structure whose shape reminded early anatomists of a seahorse. This region, found in the medial temporal lobe of each hemisphere, serves as the brain's primary memory formation center. Every conscious memory you possess passed through hippocampal circuits at some point, undergoing a complex process of encoding, consolidation, and integration with existing knowledge. The hippocampus doesn't store memories permanently, but rather serves as a temporary staging area where new experiences are processed and prepared for long-term storage in cortical networks. The hippocampus consists of several distinct subregions, each with specialized functions in memory processing. The dentate gyrus serves as the entry point for new information, containing granule cells that create sparse, distinctive patterns of activity for each experience. This region may be one of the few brain areas where new neurons continue to be born throughout adulthood, with these newly generated cells potentially contributing to the formation of new memories and the separation of similar experiences. Information flows from the dentate gyrus to the CA3 region, where powerful recurrent connections allow memories to be completed from partial cues. If you smell a particular perfume that instantly transports you back to your grandmother's house, complete with vivid details of her kitchen and the sound of her voice, you're experiencing the pattern completion abilities of CA3 neurons. These cells can reconstruct entire memory networks from fragments of sensory input, explaining how a single cue can trigger rich, detailed recollections. The CA1 region receives processed information from CA3 and serves as the hippocampus's primary output station. CA1 neurons compare incoming sensory information with stored memory patterns, detecting when current experiences differ from expectations. This novelty detection function helps focus attention on unexpected events and determines which experiences deserve to be encoded into long term memory. Routine, predictable. Events receive minimal hippocampal processing, while surprising or significant events trigger enhanced encoding that makes them more likely to be remembered. Adjacent to the hippocampus lies the amygdala, an almond shaped structure that serves as the brain's alarm system and emotional evaluation center. The amygdala constantly monitors incoming sensory information for signs of threat, opportunity, or significance, adding emotional coloring to every experience. This rapid emotional evaluation occurs largely below the threshold of consciousness, influencing behavior and decision making before rational analysis can intervene. The amygdala consists of several distinct nuclei, each specialized for different aspects of emotional processing. The lateral nucleus receives sensory input from multiple modalities and performs the initial threat assessment, responding within milliseconds to potentially dangerous stimuli. The central nucleus coordinates the physiological responses to emotional stimuli, triggering changes in heart rate, breathing, and hormone release that prepare the body for action. The basal nucleus connects to reward and motivation circuits, helping to assign positive emotional value to beneficial stimuli. Fear conditioning represents one of the amygdala's most studied functions, revealing how emotional memories form and persist. When a neutral stimulus becomes associated with danger, amygdala circuits create lasting memory traces that can trigger fear responses for years or even decades. This rapid fear learning serves crucial survival functions, allowing organisms to quickly learn which situations, people, or objects pose threats. However, the same mechanisms can also create phobias and anxiety disorders when fear conditioning occurs inappropriately or fails to extinguish when threats are no longer present. The amygdala's influence extends far beyond fear, playing crucial roles in processing positive emotions and social information. Damage to the amygdala can impair the ability to recognize emotions and facial expressions, understand social hierarchies, or form appropriate emotional attachments. The amygdala helps determine which social information deserves attention and memory encoding, influencing everything from first impressions to long term relationship formation. Connecting the hippocampus and amygdala to other brain regions are several important limbic structures that facilitate emotional memory formation and retrieval. The fornix, a large bundle of fibers, carries hippocampal output to subcortical structures, including the mammillary bodies and septal nuclei. The mammillary bodies, small spherical structures at the base of the brain, relay hippocampal information to the anterior thalamus, which in turn connects to cingulate cortex, creating a circuit essential for certain types of memory formation. The cingulate cortex, wrapped around the corpus callosum like a collar, serves multiple functions in emotional and cognitive processing. The anterior cingulate monitors conflicts between competing response tendencies and signals the need for cognitive control, while the posterior cingulate contributes to self referential thinking and autobiographical memory. The cingulate cortex represents one of the limbic system's primary interfaces with prefrontal regions responsible for executive control and decision making. The septal nuclei, located near the midline below the corpus callosum, modulate emotional responses and contribute to feelings of pleasure and reward. These structures connect to the hypothalamus and brainstem, allowing limbic emotions to influence basic physiological functions. The septal area also connects to the amygdala and hippocampus, helping to coordinate emotional and memory processing across different limbic structures. Perhaps no limbic function is more fundamental than the processing of reward and motivation, functions largely mediated by dopamine containing neurons in the ventral tegmental area and their targets in the nucleus accumbens and other forebrain structures. This reward system evaluates the motivational significance of stimuli and experiences, generating the sense of pleasure and satisfaction that guides learning and behavior. When you experience the satisfaction of completing a challenging task or the pleasure of eating a favorite food, you're experiencing the activity of these reward circuits. The reward system operates through prediction error signaling, comparing expected outcomes with actual results. When experiences exceed expectations, dopamine neurons increase their firing, creating positive reinforcement that makes similar behaviors more likely in the future. When outcomes fall short of expectations, dopamine activity decreases, creating a learning signal that can modify future behavior. This prediction error mechanism allows the brain to continuously update its models of which actions and decisions lead to desirable outcomes. Understanding reward processing helps explain the development of addictive behaviors, which essentially represent hijacking of normal motivational circuits. Drugs of abuse create artificially intense dopamine signals that overwhelm the brain's natural reward mechanisms, leading to compulsive drug seeking behavior, even when the substances no longer provide genuine pleasure or satisfaction. The same circuits that evolved to motivate survival behaviors, like eating and reproduction, can become pathologically focused on artificial stimuli. The limbic system's influence on decision making occurs through extensive connections with prefrontal cortex, creating circuits that integrate emotional and rational considerations. The orbitofrontal cortex receives direct input from limbic structures and helps translate emotional signals into behavioral choices. When making decisions, this region compares the emotional value of different options, drawing on past experiences stored in limbic memory circuits to predict the likely emotional consequences of various choices. Damage to connections between limbic structures and prefrontal cortex can lead to poor decision making despite intact intellectual abilities. Patients with such damage may score normally on intelligence tests, but make consistently poor life choices, failing to learn from negative emotional consequences or properly weigh the emotional significance of their decisions. This demonstrates how rational thought, while important, depends critically on emotional input to guide appropriate behavior. The limbic system also plays crucial roles in social behavior and emotional communication. Mirror neuron systems, found in several limbic and cortical regions, fire both when performing actions and when observing others perform similar actions. These neurons may contribute to empathy and emotional contagion, allowing us to unconsciously mirror and understand others' emotional states. The limbic system's processing of facial expressions, vocal tones, and body language provides the emotional context that makes social interaction meaningful and effective. Stress responses represent another critical limbic function coordinated primarily through the hypothalamic pituitary adrenal axis. When the amygdala detects threats, it triggers hypothalamic release of corticotropin releasing hormone, initiating a cascade that ultimately results in cortisol release from the adrenal glands. This stress response mobilizes energy and enhances attention in the short term, but chronic activation can have detrimental effects on both physical and mental health. The limbic system's memory functions extend beyond simple factual recall to include the emotional context that makes memories personally meaningful. Emotional arousal during learning enhances memory consolidation, which is why emotionally significant events are often remembered more vividly than neutral experiences. The amygdala modulates hippocampal function during emotional situations, ensuring that important experiences receive enhanced memory processing. Sleep plays a crucial role in limbic function, particularly in memory consolidation and emotional regulation. During sleep, the hippocampus replays the day's experiences, facilitating the transfer of information to long term cortical storage. The amygdala's activity during sleep may help process emotional experiences and maintain appropriate emotional responses. Sleep deprivation can impair both memory formation and emotional regulation, highlighting the importance of adequate rest for healthy limbic function. The development of limbic structures follows a protracted timeline that extends well into adolescence and early adulthood. The amygdala reaches adult like functioning relatively early, while prefrontal regions that provide cognitive control over emotional responses continue maturing into the mid 20s. This developmental mismatch may contribute to the increased emotional reactivity and risk taking behavior often observed during adolescence. Modern neuroscience has revealed that limbic functions are not localized to specific structures, but emerge from the coordinated activity of distributed networks spanning multiple brain regions. The fear network includes not only the amygdala, but also regions of the prefrontal cortex, cingulate cortex, and brainstem. The reward network encompasses dopamine neurons in the midbrain, their targets in the forebrain, and regulatory inputs from cortical areas. These network perspectives emphasize how limbic functions depend on the integration of multiple neural systems working in concert. As we prepare to explore the brainstem and diencephalon, the structures that maintain the most basic functions necessary for life, we carry with us an appreciation for how the limbic system provides the emotional and motivational foundation upon which all higher cognitive functions build. The ancient circuits we've examined continue to shape every aspect of human experience, from the formation of personal memories to the guidance of complex social behaviors. These deep brain structures remind us that even our most sophisticated thoughts and decisions remain grounded in the emotional and motivational systems that have guided behavior throughout evolutionary history. Deep within your brain, below the sophisticated networks of cortex and limbic structures, lies a collection of neural centers so fundamental to survival that their continuous operation determines the difference between life and death. These ancient regulatory structures, the brainstem and diencephalon, maintain the basic physiological processes that keep you alive moment by moment, breath by breath, heartbeat by heartbeat. While cortical regions engage in complex thought and limbic areas process emotions, these deeper structures quietly orchestrate the autonomic symphony that sustains biological existence without any conscious awareness or effort on your part. The brainstem, a narrow column of neural tissue connecting the brain to the spinal cord, represents one of the most evolutionarily conserved regions of the nervous system. Its basic organization remains remarkably similar across vertebrate species, reflecting its fundamental importance in maintaining life's essential functions. This structure houses the neural circuits that control breathing, heart rate, blood pressure, and arousal, functions so critical that even brief disruptions can have catastrophic consequences. At the base of the brainstem lies the medulla oblongata, a bulbous expansion where the spinal cord enters the skull. Despite its modest size, roughly 2 centimeters in length, the medulla contains neural circuits responsible for the most basic requirements of survival. The medullary respiratory center continuously monitors carbon dioxide levels in your blood and adjusts breathing accordingly, increasing ventilation when CO2 rises and decreasing it when levels fall. This automatic regulation occurs entirely without conscious control, allowing you to breathe naturally while sleeping, concentrating on complex tasks, or engaging in conversation. The medulla also houses the cardiovascular control center, which regulates heart rate and blood pressure through connections to the heart and blood vessels. When you stand up quickly, medullary circuits detect the drop in blood pressure and immediately trigger compensatory responses, increased heart rate, vasoconstriction, and enhanced cardiac output. These adjustments occur within seconds, preventing the dizziness or fainting that would result from inadequate brain blood flow. The precision of this control system becomes apparent only when it fails, as in certain neurological conditions that impair autonomic function. Additional medullary centers coordinate swallowing, coughing, sneezing, and vomiting, protective reflexes that prevent foreign material from entering the airway or help expel harmful substances from the body. The medulla also contains nuclei for several cranial nerves, including the hypoglossal nerve that controls tongue movements, essential for speech and swallowing, and the vagus nerve that provides parasympathetic innervation to thoracic and abdominal organs. Above the medulla lies the pons, a rounded protrusion whose name derives from the Latin word for bridge. This structure serves as a crucial relay station, containing fiber pathways that connect the cerebrum and cerebellum while housing important nuclei for cranial nerve function and arousal regulation. The pontine nuclei process information flowing from cerebral cortex to cerebellum, facilitating the coordination of voluntary movements with balance and posture. The pons contains critical components of the reticular activating system, a network of neurons that regulates consciousness and arousal. These neurons determine your level of alertness, from deep sleep through various stages of wakefulness to high arousal states. Damage to pontine arousal centers can result in coma or persistent vegetative states, highlighting their essential role in maintaining conscious awareness. The pons also participates in REM sleep generation, containing neurons that trigger the rapid eye movements and vivid dreams characteristic of this sleep stage. Several important cranial nerve nuclei reside within the pons, including the trigeminal nerve complex that processes facial sensation and controls chewing muscles, the facial nerve nucleus that controls facial expressions, and the vestibulocochlear nerve nuclei that process balance and hearing information. The pons also contains the locus coeruleus, a small nucleus containing norepinephrine-producing neurons that project throughout the brain and play crucial roles in attention, arousal, and stress responses. The midbrain, the most rostral portion of the brainstem, contains structures essential for visual and auditory reflexes, eye movement control, and motor function. The superior colliculus coordinates visual attention and eye movements, automatically directing gaze towards sudden movements or novel stimuli in the visual field. The inferior colliculus processes auditory information and contributes to sound localization, helping determine the direction and distance of sound sources. The midbrain houses the oculomotor nerve nucleus, which controls most eye movements and pupil constriction, and the trochlear nerve nucleus, which controls superior oblique muscle function. These nuclei coordinate the complex eye movements necessary for tracking objects, maintaining visual fixation, and ensuring that both eyes move together appropriately. The precision required for coordinated eye movements becomes apparent in conditions affecting these nuclei, where patients may experience double vision or difficulty focusing on objects. Perhaps most significantly, the midbrain contains the substantia nigra, a darkly pigmented region housing dopamine producing neurons that project to the basal ganglia. These neurons play crucial roles in motor control, with their degeneration causing the movement difficulties characteristic of Parkinson's disease. The midbrain also contains the red nucleus, which contributes to motor coordination, and the periaqueductal gray, a region involved in pain modulation and defensive behaviors. The reticular formation, a complex network of neurons extending throughout the brainstem, coordinates many of these basic functions while regulating arousal and consciousness. This network receives input from multiple sensory systems and projects widely throughout the brain, allowing it to modulate cortical activity based on the significance of incoming stimuli. The reticular formation determines which sensory inputs reach consciousness and which remain in the background, filtering the constant stream of sensory information to focus attention on relevant stimuli. Adjacent to the brainstem lie the structures of the diencephalon, literally meaning between brain, which serve as the interface between brainstem, limbic system, and cerebral cortex. The diencephalon includes the thalamus, hypothalamus, epithalamus, and subthalamus, each contributing specialized functions to brain regulation and information processing. The thalamus, comprising roughly 80% of the diencephalon, serves as the brain's primary relay station for sensory information. This paired structure, resembling two eggs positioned side by side, contains distinct nuclei that process different types of sensory input before relaying information to appropriate cortical areas. Visual information from the retina passes through the lateral geniculate nucleus before reaching visual cortex, while auditory information travels through the medial geniculate nucleus to auditory cortex. Somatosensory information from the body surface and deeper tissues passes through the ventral posterior nucleus before reaching somatosensory cortex. The thalamus does far more than simply relay sensory information. It actively processes and modulates the signals passing through it. Thalamic neurons can filter out irrelevant sensory input while enhancing significant stimuli, essentially controlling what information reaches conscious awareness. During sleep, thalamic circuits generate the rhythmic oscillations that characterize different sleep stages, coordinating cortical activity to facilitate memory consolidation and neural restoration. Different thalamic nuclei maintain specific connections with particular cortical areas, creating discrete thalamocortical loops that process different types of information. The ventral lateral nucleus connects with motor cortex to facilitate movement planning and execution, while the anterior nuclei connect with limbic cortex to support memory formation. The mediodorsal nucleus connects with prefrontal cortex to support executive functions and working memory. Below the thalamus lies the hypothalamus, a small but incredibly important structure that serves as the body's primary homeostatic control center. Despite weighing only about 4 grams, roughly the weight of a penny, The hypothalamus regulates body temperature, hunger, thirst, circadian rhythms, and hormonal secretion. This structure maintains the internal stability necessary for optimal cellular function, continuously monitoring and adjusting physiological parameters to keep them within narrow, acceptable ranges. The hypothalamus contains numerous distinct nuclei, each specialized for particular regulatory functions. The suprachiasmatic nucleus serves as the body's master clock, receiving direct input from the retina and coordinating circadian rhythms throughout the body. This tiny cluster of neurons, containing only about 20,000 cells, generates the approximately 24-hour rhythms that regulate sleep-wake cycles, hormone secretion, body temperature, and countless other physiological processes. The paraventricular nucleus produces hormones that regulate stress responses and water balance. When you experience stress, neurons in this nucleus release corticotropin-releasing hormone, initiating the cascade that ultimately results in cortisol release from the adrenal glands. The same nucleus produces antidiuretic hormone, which helps the kidneys retain water when the body becomes dehydrated. The arcuate nucleus monitors energy balance and regulates feeding behavior through connections with other hypothalamic regions and brainstem centers. Neurons in this region respond to hormones like leptin and ghrelin that signal energy stores and hunger, adjusting appetite and metabolism accordingly. The ventromedial nucleus contributes to satiety signaling, while the lateral hypothalamus promotes feeding behavior when energy stores become depleted. Temperature regulation represents another crucial hypothalamic function, accomplished through the preoptic area and anterior hypothalamus. These regions monitor core body temperature and initiate appropriate responses to maintain thermal homeostasis. When body temperature rises, hypothalamic circuits trigger sweating, vasodilation, and behavioral changes like seeking shade or removing clothing. When temperature falls, the same regions initiate shivering, vasoconstriction, and behaviors like adding clothing or seeking warmth. The hypothalamus exerts its regulatory influence through both neural connections and hormonal secretions. The posterior pituitary gland represents an extension of hypothalamic neural tissue, releasing hormones produced by hypothalamic neurons directly into the bloodstream. The anterior pituitary, while anatomically separate, receives regulatory hormones from the hypothalamus through a specialized blood vessel system called the hypothalamic hypophyseal portal system. This hypothalamic pituitary axis controls numerous endocrine functions throughout the body. Growth hormone releasing hormone from the hypothalamus stimulates growth hormone release from the pituitary, promoting growth and development. Thyrotropin releasing hormone triggers thyroid stimulating hormone release, regulating metabolic rate. Gonadotropin releasing hormone controls reproductive hormone secretion, coordinating sexual development and reproductive cycles. The epithalamus, located above the thalamus, contains the pineal gland and habenular nuclei. The pineal gland secretes melatonin in response to darkness, helping to regulate circadian rhythms and promote sleep. The habenular nuclei process information related to reward and aversion, contributing to learning and motivation through connections with dopamine systems in the midbrain. The subthalamus, lying below the thalamus, contains the subthalamic nucleus, which plays important roles in motor control through connections with the basal ganglia. Dysfunction of this nucleus contributes to movement disorders, and it serves as a target for deep brain stimulation treatments for Parkinson's disease. These brainstem and diencephalic structures operate through complex interconnections that coordinate their regulatory functions. The reticular formation receives input from hypothalamic centers and modulates arousal accordingly, ensuring that sleep wake cycles align with circadian rhythms. Cardiovascular and respiratory centers in the medulla respond to hypothalamic signals during stress responses, coordinating physiological changes throughout the body. The automatic nature of these regulatory functions represents one of the nervous system's most remarkable achievements. While you consciously control voluntary movements and direct attention to specific stimuli, the brainstem and diencephalon maintain the fundamental processes that sustain life without any conscious effort or awareness. This division of labor allows cortical regions to engage in complex cognitive tasks while essential functions continue uninterrupted. Understanding these structures provides insight into various neurological conditions and their symptoms. Brainstem strokes can cause dramatic symptoms despite affecting relatively small areas of neural tissue because they disrupt essential regulatory functions. Sleep disorders often involve dysfunction of brainstem and hypothalamic centers responsible for arousal and circadian regulation. Hormonal imbalances may result from hypothalamic or pituitary dysfunction affecting growth, reproduction, or stress responses. As we prepare to explore how sensory information flows through these structures to create conscious perception, we carry with us an appreciation for the fundamental regulatory functions that make all higher brain activities possible. The brainstem and diencephalon provide the stable physiological foundation upon which the elaborate constructions of sensation, emotion, and thought can build, ensuring that the basic requirements of biological existence are met while allowing consciousness to explore the complexities of experience and meaning. As photons of light enter your eyes at this very moment, they initiate one of the most remarkable transformations in biology, the conversion of electromagnetic energy into conscious visual experience. This transformation, occurring thousands of times per second, represents a masterpiece of neural engineering that transforms the simple physical properties of light waves into the rich, three-dimensional, color-filled world of visual perception. Understanding this process reveals how your brain actively constructs visual reality from fragmentary information, creating a seamless perceptual experience from what begins as mere patterns of light and shadow. The journey from light to sight begins in the retina, a thin layer of neural tissue lining the back of each eye. Despite being no thicker than a sheet of paper, the retina contains a sophisticated processing network that performs the initial stages of visual analysis before sending information to the brain. This remarkable structure represents an extension of the central nervous system, developed from the same embryonic tissue that gives rise to the brain itself. The retina's organization reflects millions of years of evolutionary refinement, with each layer serving specialized functions in visual processing. Light must pass through several layers of neural tissue before reaching the photoreceptors at the retina's back surface, an arrangement that might seem backwards, but actually serves important functional purposes. The blood vessels and supporting cells in the outer retinal layers are transparent to visible light, while this inverted organization allows the photoreceptors to be positioned adjacent to the pigment epithelium, which provides essential metabolic support and recycles visual pigments. The photoreceptors themselves come in two main varieties, rods and cones, each adapted for different aspects of vision. Rods, numbering approximately 120 million in each human retina, specialize in detecting dim light and motion. These highly sensitive cells can respond to single photons of light, making them essential for vision in low light conditions. However, rods cannot distinguish between different wavelengths of light, which is why nighttime vision appears in shades of gray rather than color. Cones, though less numerous at about 6 million per retina, provide the foundation for color vision and fine detail detection. Three types of cones exist, each containing photopigments most sensitive to different wavelengths of light. Short wavelength cones respond best to blue light. Medium wavelength cones prefer green light, and long wavelength cones are most sensitive to red light. The brain compares the relative activation of these three cone types to determine the color of objects, a process that allows humans to distinguish millions of different hues from just three basic photoreceptor types. The transformation of light into neural signals occurs through a complex biochemical cascade within the photoreceptors. When light strikes a photoreceptor, it causes a change in the shape of rhodopsin molecules embedded in the cell's membrane. This shape change triggers a cascade of chemical reactions that ultimately results in the closing of sodium channels, causing the photoreceptor to hyperpolarize rather than depolarize. This unusual response, opposite to most neural excitation, reflects the retina's sophisticated design for encoding visual information. The photoreceptors connect to a network of retinal interneurons that begin processing visual information before it leaves the eye. Bipolar cells receive input from photoreceptors and relay signals to ganglion cells, whose axons form the optic nerve. However, this simple three-neuron chain is modulated by horizontal cells and amacrine cells, which create lateral connections that enhance contrast, detect motion, and extract specific visual features. Horizontal cells connect photoreceptors across the retinal surface, creating center surround receptive fields that enhance edge detection and contrast sensitivity. When light illuminates the center of a receptive field, horizontal cells inhibit surrounding photoreceptors, creating a neural representation that emphasizes boundaries between light and dark regions. This lateral inhibition enhances our ability to detect edges and contours, the basic building blocks of object recognition. Amacrine cells provide similar lateral connections at the level of bipolar and ganglion cells, with different amacrine cell types specialized for detecting specific visual features. Some respond to directional motion, others to changes in illumination, and still others to specific patterns of light and dark. This parallel processing within the retina means that even before visual information reaches the brain, it has been decomposed into multiple streams representing different aspects of the visual scene. The ganglion cells represent the retina's output neurons, and their diverse response properties reflect the parallel processing that characterizes all subsequent visual processing. Magnocellular ganglion cells, large neurons with extensive dendritic trees, respond primarily to motion and changes in illumination. These cells sacrifice spatial resolution for temporal sensitivity, making them ideal for detecting moving objects and changes in the visual environment. Parvocellular ganglion cells, smaller neurons with more compact dendritic fields, specialize in fine spatial detail and color processing. These cells maintain high spatial resolution and can distinguish between different wavelengths of light, making them essential for reading, face recognition, and other tasks requiring detailed visual analysis. A third population, the coniocellular ganglion cells, processes blue-yellow color information and contributes to circadian rhythm regulation through connections to the hypothalamus. The axons of ganglion cells converge at the optic disc to form the optic nerve, creating a blind spot where no photoreceptors exist. Remarkably, you remain unaware of this blind spot because your brain fills in the missing information based on surrounding visual context, demonstrating the active constructive nature of visual perception. The two optic nerves meet at the optic chiasm, where axons from the nasal half of each retina cross to the opposite side of the brain, while temporal axons remain on the same side. This partial crossing creates a visual pathway organization where each hemisphere of the brain receives input from the opposite visual field. Information from the left visual field reaches the right hemisphere, while right visual field information goes to the left hemisphere. This arrangement allows for the integration of slightly different images from each eye into unified binocular vision while maintaining separate processing of different spatial regions. Most retinal ganglion cell axons terminate in the lateral geniculate nucleus of the thalamus, a six-layered structure that serves as the primary relay station for visual information traveling to the cortex. The LGN maintains the segregation of different visual processing streams, with magnocellular and parvocellular pathways terminating in different layers. The LGN also receives massive feedback from visual cortex, allowing higher level processing to influence the transmission of visual information from the very earliest stages. From the lateral geniculate nucleus, visual information travels via the optic radiations to the primary visual cortex, located in the occipital lobe at the back of the brain. This cortical area, also known as V1 or striate cortex, represents the first stage of cortical visual processing and contains approximately 200 million neurons organized into precise functional modules. The primary visual cortex maintains a detailed topographic map of the visual field, with adjacent locations in visual space represented by adjacent locations in cortex. However, this map is not uniform. The central visual field, corresponding to the fovea, where visual acuity is highest, receives disproportionately large cortical representation. This cortical magnification reflects the importance of central vision for detailed visual tasks like reading and object recognition. Within primary visual cortex, neurons are organized into columns that share similar response properties. Orientation columns contain cells that respond to lines and edges at specific orientations, while ocular dominance columns alternate between cells that respond preferentially to input from the left or right eye. This columnar organization allows the cortex to systematically analyze the visual world according to fundamental features like orientation, spatial frequency, and binocular disparity. The discovery of these response properties in visual cortex revealed that the brain doesn't simply create a passive copy of the visual world, but actively extracts specific features that are computationally useful for vision. Simple cells in V1 respond to oriented lines and edges, while complex cells respond to moving oriented stimuli regardless of their exact position within the receptive field. This progression from simple to complex response properties reflects a hierarchical processing strategy that builds increasingly sophisticated representations from elementary features. Beyond primary visual cortex, visual processing divides into two major streams that process different aspects of visual information. The ventral stream, projecting toward the temporal lobe, specializes in object recognition and identification. This what pathway processes form, color, and texture information, ultimately leading to areas that can recognize specific objects, faces, and scenes. The dorsal stream, projecting toward the parietal lobe, specializes in spatial processing and motion analysis. This where or how pathway processes information about object location, movement, and spatial relationships. The ventral stream progresses through a series of cortical areas with increasingly complex response properties. Area V4 contains neurons that respond to complex shapes and colors, while inferotemporal cortex houses neurons that respond to specific objects or object categories. Some neurons in this region respond selectively to faces, others to hands, and still others to specific types of objects like tools or animals. This categorical organization allows for rapid object recognition based on visual features. The dorsal stream processes motion and spatial information through areas like MT, middle temporal area, which contains neurons highly sensitive to motion direction and speed. Area, MST, medial superior temporal area, processes more complex motion patterns like rotation and expansion, important for analyzing self-motion and depth from motion cues. The posterior parietal cortex integrates visual information with other sensory inputs to create spatial maps used for reaching and navigation. Color vision represents one of the most elegant examples of how the brain constructs perceptual experiences from neural activity. The three types of cones provide the initial basis for color discrimination, but color perception emerges from subsequent processing that compares the outputs of different cone types. Opponent color processing in the retina and lateral geniculate nucleus creates neural channels that signal red-green, blue-yellow, and light-dark differences, forming the foundation for a rich color experience. Color constancy demonstrates the brain's remarkable ability to maintain stable color perception despite dramatic changes in illumination. A white sheet of paper appears white whether viewed under incandescent light, fluorescent light, or natural daylight, even though the wavelengths reaching your eyes differ substantially in each case. The visual system achieves this constancy by comparing the relative activation of different cone types and by using contextual information about the illumination source. Motion detection involves sophisticated neural circuits that extract velocity information from changing patterns of retinal activation. The magnocellular pathway provides the foundation for motion processing, but the computation of motion direction requires comparing signals from neurons with different spatial and temporal response properties. This processing occurs in areas like MT, where neurons respond selectively to specific directions and speeds of motion. Depth perception represents another remarkable computational achievement, allowing three-dimensional perception from two-dimensional retinal images. Binocular disparity, the slight difference between the images seen by each eye, provides precise depth information for nearby objects. Specialized neurons in visual cortex respond to specific disparities, creating a neural representation of depth that contributes to stereoscopic vision. Visual attention mechanisms allow the brain to selectively process relevant information while filtering out distractions. Attention can enhance the responses of neurons processing attended stimuli while suppressing responses to ignored stimuli, effectively controlling what information receives detailed processing. This attentional control involves interactions between visual areas and frontal and parietal cortex regions that direct attention based on goals and expectations. The constructive nature of visual perception becomes apparent in various visual illusions and perceptual phenomena. The brain constantly makes inferences about the visual world based on incomplete information, filling in missing details, and resolving ambiguities based on prior experience and contextual cues. These constructive processes usually create accurate perceptions, but they can be revealed through carefully designed stimuli that exploit the assumptions built into visual processing. Visual development requires both genetic programs and visual experience to reach full maturity. Critical periods exist during which normal visual experience is essential for proper development of binocular vision and spatial processing. Disruptions during these periods, such as those caused by cataracts or misaligned eyes, can result in permanent visual deficits, highlighting the importance of normal visual input during development. Understanding visual processing has practical implications for treating visual disorders and designing visual displays. Treatments for amblyopia, lazy eye, work by forcing the use of the weaker eye during critical developmental periods. Understanding motion processing helps in designing displays for virtual reality and understanding motion sickness. Knowledge of color processing guides the development of displays and lighting systems. As we prepare to explore how other sensory systems transform physical energy into perceptual experience, the visual system provides a template for understanding sensory processing more generally. The principles we've discovered in vision, hierarchical processing, parallel pathways, feature extraction, and constructive perception, every PAS, characterize sensory processing throughout the nervous system, revealing common strategies that the brain uses to transform the physical world into the rich tapestry of conscious experience. Within the spiral chambers of your inner ear, microscopic hair cells dance to vibrations that began as pressure waves in the air around you. These delicate sensors, no larger than a few thousandths of a millimeter, transform the mechanical energy of sound into the electrical language of the nervous system, beginning a remarkable journey that culminates in your conscious experience of music, speech, and the acoustic landscape that surrounds you. Alongside these auditory sensors, other hair cells respond to the subtlest movements of your head, maintaining your sense of balance and spatial orientation with such precision that you can walk, run, or dance without conscious effort. The auditory system faces a formidable computational challenge, extracting meaningful information from the complex mixture of sound waves that constantly bombard our ears. Every moment, your auditory system simultaneously processes hundreds of different frequencies, separates overlapping sounds from multiple sources, and extracts the temporal patterns that give meaning to speech and music. This acoustic analysis occurs with such speed and accuracy that you can instantly recognize a familiar voice in a crowded room or identify a specific instrument in a symphony orchestra. Sound begins as variations in air pressure that propagate outward from vibrating objects. These pressure waves, carrying information about frequency, amplitude, and timing, enter the outer ear and travel through the ear canal to the tympanic membrane. The middle ear, with its chain of three tiny bones, the malleus, incus, and stapes, amplifies these vibrations and transmits them to the fluid-filled chambers of the inner ear, where the real magic of auditory processing begins. The cochlea, a snail-shaped structure no larger than a pea, contains the sensory apparatus that transforms sound vibrations into neural signals. Within its spiral chambers lies the organ of Corti, a remarkable biological structure that performs frequency analysis with precision that surpasses most electronic devices. The organ of Corti rests on the basilar membrane, a flexible structure whose mechanical properties vary systematically along its length, creating a natural frequency analyzer. When sound vibrations enter the cochlea, they create traveling waves along the basilar membrane. High frequency sounds cause maximum displacement near the base of the cochlea, where the membrane is narrow and stiff, while low frequency sounds travel further toward the apex, where the membrane becomes wider and more flexible. This mechanical frequency analysis creates a tonotopic map along the cochlear length, with different locations responding optimally to different sound frequencies. The hair cells that detect these mechanical vibrations represent some of the most sophisticated sensory receptors in the body. Each hair cell contains rows of stereocilia, hair-like projections that increase in height in a stepwise fashion. When sound-induced vibrations bend these stereocilia toward the tallest row, mechanosensitive channels open, allowing ions to flow into the cell and triggering neurotransmitter release. When the stereocilia bend in the opposite direction, the channels close, reducing neurotransmitter release. Two types of hair cells populate the organ of Corti, each serving distinct functions in auditory processing. Inner hair cells, arranged in a single row along the cochlear length, serve as the primary sensory receptors. These cells connect to roughly 95% of the auditory nerve fibers and provide the main pathway for auditory information to reach the brain. Outer hair cells, arranged in 3 rows, function as biological amplifiers that enhance the sensitivity and frequency selectivity of the cochlea. The amplification provided by outer hair cells represents one of biology's most elegant engineering solutions. These cells contain contractile proteins that allow them to change length in response to electrical stimulation. When stimulated by sound at their characteristic frequency, outer hair cells contract and extend in phase with the sound vibrations, mechanically amplifying the movement of the basilar membrane. This biological amplification can increase sensitivity by up to 100-fold and sharpens frequency tuning, allowing the ear to detect sounds over an enormous dynamic range. Auditory nerve fibers carry information from the cochlea to the brainstem, maintaining the tonotopic organization. Established in the cochlea, each auditory nerve fiber responds optimally to a specific frequency, creating parallel channels that preserve frequency information throughout the auditory pathway. The temporal patterns of nerve fiber firing encode additional information about sound intensity, timing, and complex spectral features. The first central processing station for auditory information is the cochlear nucleus in the brainstem, where different cell types extract specific features from the incoming auditory signals. Bushy cells preserve precise timing information important for sound localization, while stellate cells respond to spectral features important for sound identification. T-multipolar cells extract information about sound onsets and offsets, contributing to the detection of acoustic transients. From the cochlear nucleus, auditory information travels along multiple parallel pathways that process different aspects of sound. The superior olivary complex, the first location where information from both ears converges, specializes in sound localization. Different nuclei within this complex extract different spatial cues. The medial superior olive compares the timing of sounds arriving at each ear, while the lateral superior olive compares the intensity differences between the ears. Sound localization depends on the brain's ability to analyze subtle differences in the acoustic signals reaching each ear. For low-frequency sounds, the primary cue is the difference in arrival time between the ears. Since sound travels at a finite speed, sounds originating from one side of the head reach the near ear slightly before reaching the far ear. The maximum time difference for sounds coming from directly to one side is only about 700 microseconds, yet the auditory system can detect timing differences as small as 10 microseconds. For high frequency sounds, the head creates an acoustic shadow that reduces the intensity of sounds reaching the far ear. The brain compares these intensity differences to determine sound location. The outer ear, or pinna, also provides important localization cues by filtering sounds differently depending on their direction, creating spectral patterns that help distinguish sounds coming from in front versus behind, or above versus below. The inferior colliculus in the midbrain integrates information from multiple brainstem nuclei and serves as a major convergence point for ascending auditory pathways. This structure contains neurons that respond to increasingly complex acoustic features, including specific combinations of frequencies, temporal patterns, and spatial locations. Many inferior colliculus neurons also receive input from other sensory systems, beginning the process of multisensory integration. The medial geniculate body of the thalamus serves as the primary relay for auditory information traveling to cortex. Like other thalamic nuclei, the medial geniculate body does more than simply relay information. It actively modulates the transmission of auditory signals based on attention, arousal, and cortical feedback. During sleep, thalamic circuits filter out routine sounds while remaining sensitive to significant acoustic events that might signal danger. Primary auditory cortex, located in the superior temporal gyrus, contains multiple tonotopic maps that represent different aspects of auditory processing. These maps maintain the frequency organization established in the cochlea, but add layers of complexity that extract behaviorally relevant features from acoustic signals. Neurons in primary auditory cortex respond to combinations of frequencies, specific temporal patterns, and complex spectral features that contribute to sound identification. Beyond primary auditory cortex, the auditory system divides into specialized processing streams analogous to the visual system's ventral and dorsal pathways. The ventral what stream, projecting toward the temporal lobe, specializes in sound identification and recognition. This pathway processes the spectral and temporal features necessary to distinguish different sounds, recognize speech, and appreciate music. The dorsal where stream, projecting toward the parietal lobe, specializes in spatial processing and auditory guided action. Speech processing represents one of the most sophisticated functions of the human auditory system. The acoustic patterns that constitute speech are extraordinarily complex, with multiple acoustic cues contributing to the perception of individual speech sounds. Formant frequencies, the resonant frequencies of the vocal tract, provide primary cues for vowel identification, while temporal features like voice onset time distinguish between consonants like ba and pa. The brain's remarkable ability to extract speech information from acoustic signals becomes apparent when considering the challenges of speech perception. Speech sounds vary dramatically depending on the speaker's gender, age, accent, and emotional state, yet listeners effortlessly extract the intended message. Coarticulation effects mean that the acoustic properties of individual speech sounds change depending on neighboring sounds, requiring sophisticated, context-dependent processing. Music perception involves many of the same neural mechanisms as speech processing, but emphasizes different acoustic features. Pitch relationships, rhythmic patterns, and harmonic structures create the complex perceptual experiences we associate with melody, harmony, and rhythm. The brain's sensitivity to musical structure appears early in development and shows remarkable consistency across cultures, suggesting that musical processing may exploit fundamental properties of auditory neural organization. Adjacent to the cochlea, in the inner ear, lies the vestibular system, a set of sensory organs that detect head movements and gravitational forces. This system maintains our sense of balance and spatial orientation, providing information essential for postural control, gaze stabilization, and navigation. The vestibular organs include three semicircular canals that detect rotational movements and two otolith organs that detect linear accelerations and head orientation relative to gravity. The semicircular canals are filled with fluid called endolymph and contain a sensory structure called the cupula at one end. When the head rotates, inertial forces cause the endolymph to lag behind, deflecting the cupula and bending the hair cells it contains. Each canal responds optimally to rotation in a specific plane, and the three canals in each ear are oriented to detect rotation around the three primary axes of head movement. The otolith organs, the utricle and saccule, contain hair cells embedded in a gelatinous membrane studded with calcium carbonate crystals called otoconia. These dense crystals cause the membrane to move when the head accelerates or changes orientation relative to gravity, bending the hair cells and signaling linear motion or head tilt. The utricle responds primarily to horizontal accelerations and head tilts, while the saccule detects vertical accelerations. Vestibular information travels to the brainstem via the vestibular nerve, reaching the vestibular nuclei that coordinate balance and spatial orientation. These nuclei integrate vestibular signals with visual and proprioceptive information to create a unified sense of spatial orientation and self-motion. Vestibular nuclei connect to motor systems that control posture and eye movements, ensuring that balance is maintained and vision remains stable during head movements. Understanding auditory and vestibular processing has led to important medical advances in treating hearing loss, balance disorders, and related conditions. Cochlear implants bypass damaged hair cells by directly stimulating auditory nerve fibers, while vestibular rehabilitation exercises help patients adapt to balance system damage. Research into auditory processing continues to inform the development of better hearing aids, sound localization technologies, and treatments for hearing related disorders. The vestibulo-ocular reflex represents one of the fastest and most precise motor responses in the nervous system. When the head moves, vestibular signals trigger compensatory eye movements that keep the visual world stable on the retina. This reflex can respond to head movements within 10 to 15 milliseconds, much faster than visual processing could detect retinal image motion. The precision of this reflex allows clear vision during walking, running, or other activities that involve head movement. Vestibular processing also contributes to spatial memory and navigation. The vestibular system provides information about self-motion that helps update internal maps of spatial location as we move through the environment. Patients with vestibular damage often show deficits in spatial navigation tasks, even when visual and other sensory cues remain available. The integration of auditory and vestibular information with other sensory systems creates the unified perceptual experience of a stable, three-dimensional acoustic environment. Sound localization combines auditory spatial cues with vestibular information about head orientation to maintain accurate spatial hearing as the head moves. Visual auditory integration allows sounds and sights to be properly associated in space, creating coherent multisensory objects and events. Plasticity in auditory and vestibular processing allows adaptation to changing sensory conditions throughout life. The auditory system can adjust its sensitivity and frequency tuning based on acoustic experience, while the vestibular system can recalibrate its responses to compensate for peripheral damage or changes in sensory inputs. This plasticity underlies the brain's remarkable ability to adapt to hearing aids, cochlear implants, and other assistive technologies. Understanding auditory and vestibular processing has led to important medical advances in treating hearing loss, balance disorders, and related conditions. Cochlear implants bypass damaged hair cells by directly stimulating auditory nerve fibers, while vestibular rehabilitation exercises help patients adapt to balance system damage. Research into auditory processing continues to inform the development of better hearing aids, sound localization technologies, and treatments for hearing related disorders. As we prepare to explore the intimate senses of touch, taste, and smell, the auditory and vestibular systems provide important insights into how the brain processes temporal and spatial information. The precise timing analysis required for sound localization and speech perception, and the spatial processing necessary for balance and navigation, demonstrate computational strategies that the nervous system uses across multiple sensory domains. These systems also illustrate how sensory processing depends on the integration of information across multiple modalities, setting the stage for understanding how touch, taste, and smell contribute to our rich multisensory experience of the world. Among all the senses that connect you to the world around you, three stand apart for their intimacy and directness: touch, taste, and smell. These chemical and mechanical senses require actual physical contact between your body and the environment, creating more immediate and visceral connections than the distant senses of vision and hearing. Through these intimate channels, molecules from your morning coffee directly stimulate receptors in your nose and mouth, while the pressure of these words beneath your fingertips activates thousands of touch receptors in your skin. These senses shape not only your moment to moment experience, but also your deepest emotions and most enduring memories. Touch, or somatosensation, represents perhaps the most fundamental of all sensory experiences. It is the first sense to develop in the womb and often the last to fade before death, serving as our most basic connection to physical reality. The somatosensory system encompasses not just the light touch of a feather or the pressure of a handshake, but also pain, temperature, vibration, and proprioception, EZM, the sense of body position and movement that allows you to touch your nose with your eyes closed. The skin, your body's largest organ, contains an extraordinary array of sensory receptors, each specialized for detecting different aspects of touch. Mechanoreceptors respond to physical deformation of the skin, thermoreceptors detect temperature changes, and nociceptors signal potential tissue damage through the sensation of pain. This diverse receptor population allows the somatosensory system to extract remarkably detailed information about objects and events in contact with the body. Mechanoreceptors come in several varieties, each optimized for specific types of mechanical stimulation. Meissner corpuscles, located just beneath the skin's surface, respond to light touch and low-frequency vibrations. These receptors are particularly dense in sensitive areas like fingertips and lips, where they contribute to fine tactile discrimination. Pacinian corpuscles, larger and deeper in the skin, detect high frequency vibrations and pressure changes, allowing you to feel the texture of surfaces or the vibrations of a cell phone in your pocket. Merkel discs provide information about sustained pressure and fine spatial details, contributing to the ability to read Braille or distinguish between different textures. Ruffini endings respond to skin stretch and contribute to the perception of finger position and movement. This diverse array of mechanoreceptors works together to create the rich tactile experiences that guide manual exploration and manipulation of objects. The density and sensitivity of touch receptors varies dramatically across the body surface, creating a distorted sensory map where some areas receive disproportionate neural representation. Your fingertips contain thousands of receptors per square centimeter, while your back has relatively few. This uneven distribution reflects the functional importance of different body regions for tactile exploration and manipulation. Temperature sensation depends on specialized thermoreceptors that respond to changes in skin temperature rather than absolute temperature. Warm receptors increase their firing rate when skin temperature rises above normal body temperature, while cold receptors respond to cooling. The perception of temperature involves comparing the activity of these different receptor types, which explains why the same object can feel warm or cool depending on the temperature of your skin when you touch it. Pain sensation, while often unpleasant, serves crucial protective functions by signaling actual or potential tissue damage. Nociceptors come in different varieties that respond to mechanical damage, extreme temperatures, or chemical irritants. The pain system includes both fast conducting fibers that signal sharp, localized pain and slower fibers that carry dull, burning, or aching sensations. This dual pain system allows for both immediate protective responses and longer term healing behaviors. Somatosensory information travels to the brain via two major pathways that process different aspects of touch. The dorsal column medial lemniscal pathway carries information about fine touch, vibration, and proprioception, maintaining precise spatial and temporal information about tactile stimuli. The spinothalamic pathway carries pain and temperature information, along with crude touch sensations that provide general information about stimuli contacting the body. These pathways converge in the thalamus before reaching the primary somatosensory cortex in the postcentral gyrus. The somatosensory cortex maintains a detailed map of the body's surface, with adjacent body parts represented in adjacent cortical areas. This map, often illustrated as the sensory homunculus, shows the disproportionate representation of sensitive body parts like the hands, lips, and face. Beyond primary somatosensory cortex, touch information is processed in secondary somatosensory areas that integrate tactile information with other sensory modalities and extract more complex features from tactile stimuli. These areas contribute to object recognition through touch, allowing you to identify familiar objects by their shape, texture, and weight, even without looking at them. The chemical senses of taste and smell provide more direct molecular access to the environment than any other sensory modality. These ancient senses evolved to evaluate the chemical composition of food and air, determining what is safe to eat or breathe and what should be avoided. Despite their fundamental importance for survival, taste and smell operate through remarkably similar mechanisms, both detecting specific molecules through specialized receptor proteins. Taste or gustation begins with taste buds distributed across the tongue, soft palate, and throat. Each taste bud contains 50 to 100 taste receptor cells that respond to different categories of chemical stimuli. Traditional views of taste recognized four basic categories: sweet, sour, salty, and bitter, but modern research has identified a fifth category, umami, which responds to amino acids and contributes to the savory taste of protein rich foods. Taste receptor cells use different mechanisms to detect these various chemical categories. Salt taste depends on sodium channels that allow sodium ions to directly enter the cell, while sour taste involves acid sensitive ion channels that respond to hydrogen ions. Sweet, bitter, and umami tastes involve specialized receptor proteins coupled to intracellular signaling cascades that amplify the chemical signal. The diversity of bitter taste receptors reflects the importance of detecting potentially harmful substances. Humans possess approximately 25 different bitter taste receptor types, each tuned to different classes of bitter compounds. This multiplicity allows the taste system to detect a wide range of potentially toxic substances, from plant alkaloids to spoiled food compounds. Taste information travels to the brain via three cranial nerves that innervate different regions of the mouth and throat. The facial nerve carries taste information from the front two-thirds of the tongue. The glossopharyngeal nerve innervates the back third of the tongue, and the vagus nerve carries taste information from the throat and epiglottis. These pathways converge in the brainstem before reaching the thalamus and ultimately the gustatory cortex in the insula and frontal operculum. Smell, or olfaction, represents the most direct pathway from the external environment to the brain. Olfactory receptor neurons in the nasal cavity send axons directly to the olfactory bulb without any intervening synapses, making olfaction unique among sensory systems in bypassing the thalamus. This direct connection to ancient brain regions may explain smell's powerful ability to evoke emotions and memories. The human olfactory system can distinguish thousands of different odors using only about 350 functional olfactory receptor types. Each olfactory receptor neuron expresses only one type of receptor protein, but the brain can distinguish between different odors by analyzing the patterns of activation across many different receptor types. This combinatorial coding allows a relatively small number of receptor types to signal an enormous number of different chemical compounds. Olfactory receptor neurons are unique in the nervous system because they are continuously replaced throughout life. These neurons extend from the nasal cavity through the skull to synapse directly with neurons in the olfactory bulb. The olfactory bulb processes and relays smell information to various brain regions, including the piriform cortex, amygdala, and hippocampus. The direct connections between olfactory areas and limbic structures may explain why smells can so powerfully trigger emotional responses and memories. The scent of a particular perfume might instantly transport you back to a specific time and place, complete with associated emotions and detailed memories. This phenomenon, sometimes called the Proust effect, after the famous literary description of memory triggered by the smell of madeleines, demonstrates the intimate connection between olfaction and memory systems. The integration of taste and smell creates the complex perceptual experience known as flavor. What we commonly think of as taste actually depends heavily on smell, which is why food loses much of its flavor when you have a cold that blocks nasal airflow. Retronasal olfaction, the detection of food odors that reach the nose through the back of the throat during eating, contributes significantly to flavor perception. The chemical senses also include the trigeminal system, which detects chemical irritants through pain and temperature receptors in the mouth and nose. The burning sensation of chili peppers, the cooling effect of menthol, and the pungency of ammonia all activate trigeminal receptors rather than true taste or smell receptors. These sensations add important dimensions to flavor experience and serve protective functions by signaling potentially harmful chemical exposures. Sensory adaptation plays important roles in touch, taste, and smell, allowing these systems to maintain sensitivity to changes while avoiding saturation from constant stimulation. Touch receptors adapt to sustained pressure, which is why you stop feeling your clothes shortly after putting them on. Taste and smell receptors also adapt to continued exposure, reducing sensitivity to familiar flavors and odors while maintaining responsiveness to new chemical stimuli. Individual differences in sensory sensitivity create substantial variation in touch, taste, and smell experiences. Some people, called supertasters, have higher densities of taste buds and experience taste more intensely than others. Genetic variations in olfactory receptors mean that some odors smell completely different to different people, while others cannot detect certain odors at all. The development and aging of these sensory systems follow different trajectories. Touch sensitivity is present from birth and remains relatively stable throughout life, though aging can reduce the density of some receptor types. Taste sensitivity peaks in childhood and gradually declines with age, while smell sensitivity often shows more dramatic age-related changes that can significantly impact quality of life and nutrition. Pain represents a special case among the somatosensory modalities because of its complex interactions with emotional and cognitive systems. The experience of pain involves not just the detection of potentially harmful stimuli, but also emotional and cognitive evaluation of the threat they represent. This is why the same stimulus can be experienced as more or less painful depending on context, attention, and emotional state. Chronic pain conditions illustrate how plastic changes in somatosensory processing can lead to persistent pain even in the absence of ongoing tissue damage. These conditions often involve sensitization of pain pathways that amplifies normal sensory signals, creating pain experiences that no longer serve protective functions. Understanding these mechanisms has led to new approaches for treating chronic pain that target central nervous system processing rather than just peripheral tissue damage. The integration of touch, taste, and smell with other sensory modalities creates rich multisensory experiences that enhance perception and guide behavior. Visual information about food appearance influences taste perception, while touch information contributes to the perception of food texture and temperature. The sound of food crunching combines with its taste, smell, and texture to create the complete eating experience. These intimate senses also play crucial roles in social interaction and emotional communication. Touch conveys emotional information through handshakes, hugs, and other forms of physical contact. Taste and smell contribute to social bonding through shared meals and the recognition of family members. The loss of these senses can have profound impacts on social relationships and emotional well-being. On social relationships and emotional well-being. As we prepare to explore how the brain controls movement and action, the intimate senses remind us that perception is not a passive process of receiving information from the environment. Instead, these senses reveal how the nervous system actively explores and evaluates the world through direct physical and chemical contact. The integration of information from these diverse sensory channels creates the unified perceptual experience that guides our interactions with the physical and social world around us. The transformation of intention into action represents one of the nervous system's most remarkable achievements. As you turn the page or adjust your position while reading, your brain orchestrates an intricate dance of neural activity that translates abstract thoughts into precise muscle contractions. This process involves multiple brain regions working in concert to plan movements, coordinate timing, integrate sensory feedback, and adapt motor commands based on changing circumstances. Understanding motor control reveals how the brain bridges the gap between mind and body, transforming the electrical patterns of neural activity into the physical movements that allow us to interact with the world. Motor control begins long before muscles contract, with neural processes that plan and prepare movements based on goals and environmental conditions. The motor system operates through a hierarchical organization where higher level areas establish movement goals and strategies, while lower level areas implement the detailed commands necessary to achieve those goals. This hierarchical control allows for both flexible planning and precise execution, enabling everything from the gross movements needed to walk across a room to the fine motor control required to thread a needle. The primary motor cortex, located in the precentral gyrus of the frontal lobe, serves as the brain's primary output for voluntary movement commands. This region contains a systematic map of the body's musculature, with different cortical areas controlling different muscle groups. The motor homunculus, like its sensory counterpart, shows disproportionate representation of body parts requiring fine motor control. The areas controlling hand and facial muscles occupy large cortical territories, reflecting the precision required for manipulation and expression. Motor cortex neurons fire in patterns that encode multiple aspects of movement, including direction, force, and timing. Population vector algorithms allow researchers to decode intended movement direction from the collective activity of motor cortex neurons, a principle that underlies brain-computer interfaces that enable paralyzed patients to control robotic arms or computer cursors through thought alone. These technologies demonstrate the direct relationship between neural activity patterns and intended movements. The motor cortex does not operate in isolation, but receives extensive input from other brain regions involved in movement planning and control. The premotor cortex, located anterior to primary motor cortex, contributes to movement preparation and the selection of appropriate motor programs. This region integrates sensory information about the environment with motor intentions, helping to plan movements that are appropriate for current circumstances. The supplementary motor area specializes in the planning and coordination of complex, self-initiated movements. This region becomes active during the preparation of movement sequences and plays important roles in bimanual coordination tasks that require the two hands to work together. Damage to the supplementary motor area can impair the ability to initiate movements spontaneously while leaving the ability to respond to external cues relatively intact. The posterior parietal cortex contributes crucial spatial information for motor control, integrating visual and proprioceptive information to specify target locations and guide reaching movements. This region transforms sensory information about object locations from eye-centered coordinates into the body-centered coordinates needed for motor control. Neurons in posterior parietal cortex encode both the sensory properties of movement targets and the motor intentions directed toward them. The basal ganglia, a collection of subcortical nuclei, including the caudate nucleus, putamen, globus pallidus, and substantia nigra, play essential roles in motor control through their influence on cortical motor areas. The basal ganglia receive input from throughout the cerebral cortex and send output back to motor areas via the thalamus, creating loops that modulate cortical activity. These circuits are particularly important for the selection and initiation of movements, helping to determine which of many possible actions should be executed at any given moment. The basal ganglia operate through a complex pattern of excitatory and inhibitory connections that normally suppress unwanted movements while facilitating desired actions. The direct pathway through the basal ganglia promotes movement by reducing the inhibition that normally suppresses motor cortex activity. The indirect pathway provides a brake on movement by increasing this inhibition. The balance between these pathways determines whether movements are initiated smoothly or suppressed appropriately. Dopamine producing neurons in the substantia nigra modulate basal ganglia function and play crucial roles in movement, initiation, and motor learning. The loss of these dopamine neurons in Parkinson's disease leads to the characteristic movement difficulties, including slowness, rigidity, and tremor. Understanding basal ganglia function has led to effective treatments for Parkinson's disease, including medications that replace lost dopamine and deep brain stimulation that modulates abnormal neural activity patterns. The cerebellum, despite comprising only 10% of the brain's volume, contains over half of all neurons in the nervous system. This remarkable structure serves as the brain's movement coordinator, receiving copies of motor commands from the cerebral cortex and comparing them with sensory feedback about actual movement outcomes. The cerebellum detects discrepancies between intended and actual movements, generating error signals that improve motor performance through learning. Cerebellar circuitry exhibits a highly regular organization that appears optimized for motor learning. Purkinje cells, the cerebellum's output neurons, receive two types of input: parallel fibers that convey information about movement context, and climbing fibers that signal movement errors. When climbing fibers fire simultaneously with parallel fibers, they modify the strength of parallel fiber synapses, creating a cellular mechanism for motor learning that can adapt movements based on experience. Different regions of the cerebellum contribute to different aspects of motor control. The vestibulocerebellum helps maintain balance and coordinate eye movements, receiving input from vestibular organs and visual areas. The spinocerebellum modulates ongoing movements by comparing intended movements with sensory feedback about actual performance. The cerebrocerebellum, the largest division, contributes to movement planning and motor learning, particularly for skilled voluntary movements. Cerebellar damage produces characteristic movement disorders that illustrate its normal functions. Ataxia, the incoordination resulting from cerebellar dysfunction, affects both gross and fine motor control. Patients may show intention tremor that worsens as they approach targets, dysmetria that causes movements to overshoot or undershoot their goals, and dysdiadochokinesia, difficulty performing rapid, alternating movements. These symptoms demonstrate the cerebellum's roles in movement timing, coordination, and adaptation. Motor learning represents a fundamental aspect of motor control that allows the nervous system to improve performance through experience. Different types of motor learning involve different neural mechanisms and brain regions. Adaptation learning, such as adjusting to new visual motor relationships or force fields, primarily involves the cerebellum. Skill learning, such as learning to play a musical instrument or type on a keyboard, involves multiple brain regions, including motor cortex, basal ganglia, and cerebellum. The process of motor skill acquisition typically progresses through distinct stages. The cognitive stage involves conscious attention to movement components and frequent errors. The associative stage shows improved consistency as movements become more automatic. The autonomous stage achieves skilled performance with minimal conscious control and high resistance to interference. These stages involve shifts in the brain networks controlling movement, with decreased involvement of attention and cognitive control areas as skills become automated. Sensory feedback plays crucial roles in motor control, providing information about movement outcomes that enables both immediate corrections and long-term learning. Visual feedback about hand position helps guide reaching movements, while proprioceptive feedback about joint angles and muscle lengths provides information about body configuration. Tactile feedback contributes to grip force, control, and object manipulation. The integration of sensory feedback with motor commands occurs at multiple levels of the nervous system. Spinal reflexes provide the fastest corrections, responding to unexpected perturbations within 30 to 50 milliseconds. Transcortical reflexes, involving pathways through the cerebral cortex, provide more sophisticated corrections that can be modified based on task context and goals. Voluntary corrections occur with longer delays, but allow for complex adaptive responses to environmental changes. Internal models represent sophisticated neural mechanisms that the brain uses to predict the sensory consequences of motor commands. Forward models predict the sensory effects of movements, allowing the motor system to compensate for delays in sensory feedback and to distinguish between self-generated and externally imposed sensory changes. Inverse models compute the motor commands needed to achieve desired movements, essentially solving the problem of transforming movement goals into appropriate muscle activations. These internal models enable remarkable feats of motor control, such as the ability to catch a ball despite the delays inherent in visual processing and motor execution. The brain predicts where the ball will be based on its trajectory and generates motor commands that will position the hand at the right place and time. This predictive control is essential for skilled movements in dynamic environments. Motor control also exhibits remarkable plasticity that allows adaptation to changing conditions throughout life. The motor system can compensate for injury, adapt to new tools or environmental constraints, and learn new skills even in adulthood. This plasticity involves changes in synaptic strength, cortical map organization, and the recruitment of alternative neural pathways. Following brain injury, motor recovery often involves both restoration of function in damaged areas and compensation by alternative neural pathways. Rehabilitation techniques that emphasize intensive, task-specific practice can promote cortical reorganization that supports functional recovery. Understanding the neural basis of motor plasticity has led to improved rehabilitation strategies, including constraint-induced movement therapy and robotic-assisted training. The development of motor control follows predictable sequences that reflect the maturation of different brain regions and neural pathways. Motor milestones such as reaching, grasping, sitting, and walking emerge as the necessary neural circuits come online. The prolonged development of motor skills in humans, extending well into adolescence for complex skills, reflects the extensive learning required to master sophisticated movement patterns. Motor control disorders provide important insights into normal motor function. Dystonia involves involuntary muscle contractions that cause abnormal postures or movements. Chorea produces rapid, dance like movements that reflect dysfunction in basal ganglia circuits. These conditions demonstrate how disruptions in specific neural circuits can produce characteristic movement abnormalities. The conscious control of movement represents only a small fraction of motor system activity. Most motor control occurs automatically below the threshold of consciousness. This automatic control allows attention to be directed toward high level goals, while lower level systems handle the details of movement execution. The ability to perform complex movements without conscious control represents a major evolutionary advantage that frees cognitive resources for other tasks. Modern research techniques, including brain imaging, single cell recordings, and computational modeling, continue to reveal new aspects of motor control. Brain computer interfaces demonstrate direct relationships between neural activity and movement intentions. Optogenetic techniques allow precise control of neural activity in animal models, revealing causal relationships between neural circuits and behavior. As we prepare to explore the higher cognitive functions that emerge from this foundation of perception and action, motor control provides essential insights into how the brain translates abstract intentions into concrete actions. The motor system demonstrates principles of hierarchical organization, feedback control, and adaptive learning that characterize neural function more broadly. Understanding how the brain controls movement reveals fundamental mechanisms that enable all goal-directed behavior and voluntary action. Perhaps no cognitive ability distinguishes humans more dramatically from other species than our capacity for language. As you process these words, your brain performs computations of staggering complexity, transforming abstract symbols into meaningful concepts, retrieving memories associated with each word, and constructing an understanding that transcends the simple sum of individual meanings. This remarkable ability emerges from specialized neural networks that have evolved to support the infinite creativity of human language, allowing us to express novel thoughts, share complex ideas across generations, and coordinate sophisticated social behaviors that define human civilization. Language represents far more than a simple communication system. While other species possess sophisticated ways of conveying information about immediate environmental conditions or emotional states, human language exhibits unique properties that enable unlimited expression. We can discuss abstract concepts that exist only in our minds, describe events from the distant past or imagined future, and create entirely fictional worlds through narrative. This generative capacity allows language to serve not just as a tool for communication, but as a medium for thought itself. The neural foundations of language were first revealed through careful study of patients with brain damage that selectively impaired language abilities. In the 1,860 seconds, French physician Paul Broca described patients who could understand speech but could produce only limited, effortful utterances. Post-mortem examination revealed damage to a specific region in the left frontal lobe, now known as Broca's area. A decade later, German neurologist Karl Wernicke described patients with the opposite pattern, fluent but meaningless speech accompanied by poor comprehension, associated with damage to the left temporal lobe in what became known as Wernicke's area. These early discoveries established the fundamental principle that language processing is lateralized, predominantly occurring in the left hemisphere in most individuals. They also revealed that language involves multiple specialized components that can be selectively impaired by focal brain damage. However, modern neuroscience has revealed that the classical Broca-Wernicke model, while historically important, represents a significant oversimplification of how the brain actually processes language. Contemporary understanding recognizes that language processing involves extensive networks spanning multiple brain regions, each contributing specialized functions to the complex task of linguistic communication. Rather than being localized to just two areas, language processing engages regions throughout the left hemisphere and increasingly involves bilateral networks for more complex linguistic tasks. Broca's area, more precisely defined as areas 44 and 45 inches, the inferior frontal gyrus, plays crucial roles in speech production, grammatical processing, and syntactic analysis. This region coordinates the complex motor sequences required for articulate speech, involving precise timing of respiratory, laryngeal, and articulatory movements. Beyond motor control, Broca's area processes grammatical relationships between words, enabling the understanding and production of complex syntactic structures that convey meaning through word order and grammatical markers. Wernicke's area, located in the posterior superior temporal gyrus, specializes in lexical processing and semantic comprehension. This region accesses stored word meanings and integrates them into coherent interpretations of sentences and discourse. Wernicke's area connects extensively with other temporal lobe regions involved in conceptual knowledge, allowing linguistic input to activate rich networks of associated meanings and memories. The connection between these regions occurs through the arcuate fasciculus, a major white matter pathway that allows rapid communication between frontal and temporal language areas. This connection enables the integration of semantic processing in temporal regions with grammatical analysis in frontal areas, supporting the complex coordination required for both language comprehension and production. Modern neuroimaging has revealed additional components of the language network that contribute specialized functions. The angular gyrus in the inferior parietal lobe serves as a convergence zone where linguistic, conceptual, and spatial information integrate. This region plays important roles in reading comprehension, metaphor processing, and the integration of language with other cognitive domains. The middle temporal gyrus contains areas specialized for different aspects of word processing. The posterior middle temporal gyrus responds to phonological properties of words, e.i., their sound patterns, while more anterior regions process semantic properties, e.i., their meanings. This functional division allows the brain to simultaneously access both the sound and meaning of words during language processing. The inferior frontal cortex, extending beyond classical Broca's area, contributes to various aspects of language processing, including working memory for linguistic information, cognitive control during sentence processing, and the resolution of ambiguous meanings. These regions help maintain linguistic information in active memory while complex sentences are being processed and resolve conflicts when words or sentences have multiple possible interpretations. Speech production involves a remarkable coordination of neural systems that transform abstract linguistic intentions into precisely timed motor commands. The process begins with conceptual planning in regions that access semantic knowledge and select appropriate words to express intended meanings. These semantic representations must then be converted into phonological forms, is the sound patterns of words, before being translated into motor commands for articulation. Phonological processing involves areas in the superior temporal gyrus that maintain sound based representations of words and syllables. These regions work closely with inferior frontal areas that sequence phonological units into fluent speech output. The coordination between these areas enables the rapid automatic process of speaking that typically occurs at rates of 2 to 3 words per second with remarkable precision. Motor aspects of speech production involve extensive cortical and subcortical networks that control respiratory, laryngeal, and articulatory movements. Primary motor cortex regions controlling speech muscles show specialized organization that reflects the precise coordination required for articulate speech. Subcortical structures, including the basal ganglia and cerebellum, contribute to the timing and sequencing of speech movements. Language comprehension involves the rapid integration of multiple types of linguistic information as words are heard or read. Phonological processing extracts speech sounds from the acoustic signal, while lexical processing accesses stored word meanings. Syntactic processing analyzes grammatical relationships between words, and semantic integration combines word meanings into sentence level interpretations. This processing occurs with remarkable speed, with the brain beginning to access word meanings within 200 milliseconds of hearing a word. Event-related potential studies reveal distinct neural responses to different types of linguistic violations, including phonological mismatches, syntactic errors, and semantic anomalies. These responses demonstrate that the brain rapidly and automatically processes multiple levels of linguistic structure simultaneously. Reading represents a relatively recent cultural invention that appropriates neural circuits originally evolved for other functions. The visual word form area in the left occipitotemporal cortex specializes in recognizing written words and letter strings, developing through literacy experience to become highly sensitive to orthographic patterns. This region connects to the broader language network, allowing written words to access the same semantic and phonological representations used for spoken language. The dual route model of reading proposes that written words can be processed through two pathways: a direct route that connects visual word forms to meanings, and an indirect route that converts letters to sounds before accessing meanings. Skilled readers use both routes, with the direct route supporting rapid recognition of familiar words and the indirect route enabling pronunciation of unfamiliar words through phonological decoding. Writing involves additional motor and spatial processing systems that coordinate with language networks to produce written expression. The planning and execution of writing movements involves premotor and motor cortical areas, while the spatial organization of written text engages parietal cortex regions involved in spatial processing. These systems must coordinate with language areas to translate linguistic intentions into written form. Bilingual and multilingual individuals demonstrate remarkable neuroplasticity in organizing multiple language systems. Brain imaging studies reveal that different languages can show both overlapping and distinct patterns of activation, depending on factors like age of acquisition, proficiency, and similarity between languages. Early bilinguals often show more overlapping activation patterns, while late bilinguals may maintain more distinct neural representations for different languages. Language acquisition in children reveals the remarkable capacity of the developing brain to extract linguistic structure from environmental input. Children begin producing their first words around 12 months and show explosive vocabulary growth during the second year of life. Grammatical development follows predictable stages, with children initially producing single words, then two-word combinations, and gradually mastering complex syntactic structures. Critical period effects in language acquisition suggest that certain aspects of language are most easily acquired during specific developmental windows. Children who acquire language during the normal developmental period achieve native like proficiency, while those who experience language deprivation show lasting deficits. However, the concept of a strict critical period has been modified by evidence that adults retain considerable capacity for language learning, though they rarely achieve native like proficiency in all aspects of a second language. The relationship between language and thought represents one of the most enduring questions in cognitive science. The Sapir-Whorf hypothesis suggests that language shapes thought, with different languages potentially leading to different ways of conceptualizing the world. While strong versions of this hypothesis have been largely rejected, research continues to reveal subtle ways in which language can influence cognitive processes. Studies of color perception in speakers of different languages suggest that color categories can influence color discrimination and memory. Speakers of languages with more basic color terms show enhanced discrimination around color boundaries, while spatial language differences correlate with spatial reasoning abilities. These findings suggest that while language does not determine thought, it can influence certain aspects of cognitive processing. Language disorders provide important insights into the neural organization of language. Aphasia following stroke can selectively impair different aspects of language processing, revealing the functional specialization of different brain regions. Developmental language disorders demonstrate how genetic factors can specifically affect language acquisition while leaving other cognitive abilities relatively intact. Stuttering involves disruptions in the neural networks controlling speech fluency, with neuroimaging studies revealing differences in white matter organization and functional connectivity in individuals who stutter. Treatment approaches that modify neural activity through feedback or brain stimulation demonstrate the plasticity of language networks and their potential for rehabilitation. The evolution of language represents one of the most significant developments in human cognitive evolution. While other species show sophisticated communication abilities, none exhibit the open-ended generative capacity that characterizes human language. The evolutionary origins of language likely involved gradual changes in vocal tract anatomy, neural organization, and cognitive abilities that collectively enabled the emergence of symbolic communication. Comparative studies of communication in other species provide insights into the evolutionary foundations of language. While great apes can learn to use symbols in laboratory settings, they do not spontaneously develop symbolic communication in the wild. This suggests that human language involves unique cognitive and neural adaptations that are not present in our closest relatives. The social aspects of language processing involve specialized neural networks that integrate linguistic information with social context. Understanding irony, metaphor, and other forms of non-literal language requires integrating linguistic content with knowledge about speaker intentions and social relationships. These processes involve regions including the right hemisphere, which contributes to discourse processing and pragmatic understanding. Language processing also shows remarkable individual differences that reflect variations in neural organization, cognitive abilities, and experience. Some individuals show exceptional language abilities, such as simultaneous interpreters who can process and translate language in real time. Others may have specific language difficulties despite normal intelligence, demonstrating the separability of language from other cognitive abilities. As we prepare to explore the executive functions and consciousness that coordinate all aspects of cognitive processing, language provides crucial insights into the computational principles underlying human cognition. The hierarchical organization of language processing, from sounds to words to sentences to discourse, parallels the hierarchical organization found throughout the nervous system. The rapid, automatic nature of language processing demonstrates the remarkable efficiency that can be achieved through specialized neural circuits, while the creative, generative capacity of language reveals the open ended computational potential of neural networks. At the apex of the brain's hierarchical organization sits the prefrontal cortex, a region that orchestrates the complex symphony of human cognition with the precision of a master conductor. This area, which comprises nearly 1/3 of the human cerebral cortex, serves as the brain's executive headquarters, coordinating attention, planning future actions, making decisions, and maintaining the flexible cognitive control that allows humans to adapt to an ever changing world. Within these neural networks also emerges perhaps the most mysterious aspect of brain function: consciousness itself, the subjective experience of awareness that transforms the brain's information processing into the rich, unified experience of being. The prefrontal cortex represents the most recent evolutionary addition to the mammalian brain, reaching its greatest development in humans. This expansion reflects the increasing complexity of cognitive demands faced by our species, from navigating intricate social relationships to planning long term goals that extend far beyond immediate needs. The prefrontal cortex provides the cognitive flexibility that allows humans to override automatic responses, consider multiple alternatives, and pursue abstract goals that may conflict with immediate impulses. Executive function encompasses a suite of cognitive abilities that enable goal directed behavior in complex environments. These functions include working memory, which maintains relevant information in an active state, cognitive flexibility, which allows switching between different mental sets or strategies, and inhibitory control, which suppresses inappropriate responses while facilitating appropriate ones. Together, these abilities enable the purposeful, organized behavior that characterizes mature human cognition. Working memory represents perhaps the most fundamental executive function, providing a mental workspace where information can be temporarily maintained and manipulated. Unlike simple short term memory, which passively holds information for brief periods, working memory actively processes information while keeping it accessible for ongoing cognitive operations. When you perform mental arithmetic, follow complex directions, or engage in abstract reasoning, you rely on working memory to maintain relevant information while processing it. The neural basis of working memory involves sustained neural activity in prefrontal cortex networks that maintain information-specific firing patterns throughout delay periods. Different prefrontal regions specialize in maintaining different types of information. Dorsolateral prefrontal cortex excels at maintaining spatial and abstract information, while ventrolateral prefrontal cortex specializes in maintaining object and verbal information. These regions work together to create the flexible mental workspace that supports complex reasoning. Working memory capacity varies significantly among individuals and shows systematic changes throughout development and aging. Children show gradual improvements in working memory capacity that parallel the maturation of prefrontal cortex connections, while older adults often experience working memory declines that correlate with age related changes in prefrontal structure and function. These individual differences in working memory capacity predict performance on a wide range of cognitive tasks, from academic achievement to fluid intelligence. Attention represents another crucial executive function that determines which information receives processing priority among the constant stream of sensory inputs, memories, and internal thoughts competing for cognitive resources. The prefrontal cortex exerts top down control over attention, directing focus toward goal relevant information while filtering out distractions. This attentional control allows humans to maintain focus on important tasks despite the presence of competing stimuli. The attention networks of the brain include alerting systems that maintain vigilant readiness, orienting systems that direct attention to specific locations or features, and executive control systems that resolve conflicts between competing inputs. The anterior cingulate cortex plays a crucial role in monitoring conflicts and signaling the need for cognitive control, while dorsolateral prefrontal cortex implements the actual control processes that resolve attention conflicts. Selective attention operates through both enhancement of relevant information and suppression of irrelevant information. Brain imaging studies reveal that attending to specific visual locations or features increases neural activity in corresponding sensory areas, while attention away from locations or features decreases activity in those same areas. This bidirectional control allows the brain to optimize information processing by amplifying important signals while reducing neural noise from irrelevant sources. Sustained attention, the ability to maintain focus over extended periods, involves different neural mechanisms than selective attention. The right hemisphere, particularly right frontal and parietal areas, shows specialization for sustained attention tasks. Deficits in sustained attention characterize various neurological and psychiatric conditions, including attention deficit hyperactivity disorder and traumatic brain injury. Inhibitory control enables the suppression of automatic or prepotent responses in favor of more appropriate behaviors. This cognitive control function develops gradually throughout childhood and adolescence, reflecting the prolonged maturation of prefrontal cortex circuits. The classic Stroop task, where participants must name the color of words while ignoring the word meaning, demonstrates inhibitory control by requiring suppression of the automatic reading response. Different types of inhibitory control involve different neural circuits within the prefrontal cortex. Response inhibition, stopping ongoing motor responses, involves the right inferior frontal gyrus and its connections to motor areas. Interference control, resolving conflicts between competing response tendencies, engages anterior cingulate and dorsolateral prefrontal cortex. Cognitive inhibition, suppressing irrelevant thoughts or memories, involves dorsolateral and ventrolateral prefrontal regions. The development of inhibitory control follows a protracted timeline that extends into early adulthood. Young children show limited ability to suppress prepotent responses, leading to characteristic behaviors like difficulty waiting for rewards or following complex rules. Adolescents show improved inhibitory control compared to children, but still demonstrate limitations, particularly in emotionally charged situations where limbic system activation can overwhelm prefrontal control. Decision making represents a complex executive function that integrates information from multiple sources to select among alternative courses of action. The prefrontal cortex evaluates options based on their potential outcomes, considering both immediate and long term consequences. This evaluation process involves orbitofrontal cortex, which assigns values to different options, and dorsolateral prefrontal cortex, which compares options and implements decisions. The neural basis of decision making involves interactions between prefrontal cortex and limbic structures that provide emotional and motivational input. The orbitofrontal cortex integrates sensory information about potential rewards with emotional significance provided by limbic areas, creating value representations that guide choice behavior. The anterior cingulate cortex monitors decision outcomes and adjusts future choices based on prediction errors. Decision making under uncertainty involves additional neural mechanisms that assess risk and manage ambiguity. The prefrontal cortex maintains probabilistic representations of outcome likelihoods and adjusts these representations based on experience. Individual differences in risk tolerance correlate with differences in prefrontal and limbic activity patterns during decision tasks. Planning involves the generation and evaluation of action sequences aimed at achieving future goals. This executive function requires the ability to mentally simulate future states, evaluate alternative action sequences, and select plans that are likely to succeed. The prefrontal cortex supports planning through its connections with motor areas that enable action simulation and limbic areas that evaluate goal states. Hierarchical planning involves the decomposition of complex goals into manageable subgoals and action sequences. The prefrontal cortex maintains goal hierarchies that organize behavior across multiple timescales, from immediate actions to long-term objectives. This hierarchical organization allows humans to pursue complex, multi-step goals while maintaining flexibility to adapt plans when circumstances change. Planning deficits following prefrontal damage reveal the importance of this region for goal-directed behavior. Patients with frontal lobe lesions may show preserved basic cognitive abilities but demonstrate profound difficulties in planning and organizing their daily activities. These deficits highlight the distinction between having knowledge and being able to use it effectively in pursuit of goals. Cognitive flexibility enables adaptation to changing task demands and environmental circumstances. This executive function involves the ability to switch between different mental sets, update working memory contents, and modify behavior based on feedback. The prefrontal cortex supports cognitive flexibility through its role in maintaining task representations and implementing task switching. Set-shifting tasks require participants to switch between different categorization rules or response mappings, revealing the neural mechanisms of cognitive flexibility. The prefrontal cortex shows increased activation during task switches, particularly when switches are unexpected or require overriding strong response tendencies. Aging and various neurological conditions can impair cognitive flexibility, leading to perseverative behaviors and difficulties adapting to changing circumstances. The emergence of consciousness from neural activity represents one of the deepest mysteries in neuroscience. While the brain processes vast amounts of information continuously, only a small fraction reaches conscious awareness. Understanding how neural activity gives rise to subjective experience requires examining both the neural correlates of consciousness and the computational functions that consciousness serves. Global workspace theory proposes that consciousness arises when information becomes globally accessible across multiple brain systems. According to this view, conscious percepts result from the global broadcasting of information that was initially processed locally in specialized brain areas. The prefrontal cortex plays a crucial role in this global integration, maintaining and manipulating information that becomes conscious. Integrated information theory offers an alternative perspective, suggesting that consciousness corresponds to integrated information in neural networks. This theory proposes that conscious systems must have high levels of both differentiation, allowing distinct states, and integration, binding information into unified experiences. The thalamocortical system, with its rich interconnections and dynamic activity patterns, appears well suited to generate integrated information. The neural correlates of consciousness include specific patterns of brain activity that distinguish conscious from unconscious processing. Conscious perception is associated with sustained activity in sensory areas, global connectivity between brain regions, and specific oscillatory patterns that may bind distributed neural activity into coherent representations. The prefrontal cortex and parietal cortex show particularly strong activation during conscious processing tasks. Attention and consciousness, while related, represent distinct aspects of information processing. Attention can operate without consciousness, as demonstrated by studies of subliminal priming and blind sight. Conversely, some information may reach consciousness without focused attention, as shown by studies of change blindness and inattentional blindness. These dissociations suggest that attention and consciousness involve partially overlapping but distinct neural mechanisms. The contents of consciousness show systematic biases that reflect the computational priorities of neural processing. Conscious perception tends to favor coherent, stable interpretations over fragmented or rapidly changing ones. This bias toward stability may reflect the function of consciousness in maintaining coherent behavioral goals and plans across time. Self-awareness represents a sophisticated form of consciousness that involves the ability to reflect on one's own mental states and processes. The prefrontal cortex, particularly the medial prefrontal regions, plays crucial roles in self-referential processing and metacognition. These areas monitor ongoing cognitive processes and maintain representations of self-knowledge that guide behavior. Metacognition, or thinking about thinking, enables humans to evaluate their own cognitive processes and adjust strategies accordingly. The prefrontal cortex supports metacognitive monitoring through its connections with other brain regions, allowing assessment of confidence, accuracy, and strategy effectiveness. Individual differences in metacognitive abilities predict learning outcomes and adaptive behavior. The development of executive functions in consciousness follows predictable patterns that reflect brain maturation. Young children show limited executive control and self-awareness, with gradual improvements continuing through adolescence. The protracted development of prefrontal cortex, with some connections not reaching full maturity until the mid-20s, underlies these extended developmental changes. Executive function deficits characterize numerous neurological and psychiatric conditions. Attention deficit hyperactivity disorder involves impairments in attention and inhibitory control. Autism spectrum disorders often include difficulties with cognitive flexibility and working memory. Schizophrenia frequently involves executive dysfunction that contributes to functional impairments beyond primary symptoms. Understanding executive functions has led to interventions aimed at improving cognitive control abilities. Cognitive training programs target specific executive functions through repeated practice, while mindfulness interventions may enhance attention and awareness. Physical exercise and aerobic fitness also show benefits for executive function, particularly in older adults and children. The prefrontal cortex and its executive functions represent the pinnacle of neural evolution, enabling the flexible, goal directed behavior that characterizes human cognition. These systems coordinate the vast array of neural processes we have explored, from basic sensory processing to complex motor control, creating the unified, purposeful behavior that defines human intelligence. As we turn to examine how the brain forms and retrieves the memories that inform executive decisions, we carry with us an appreciation for the remarkable neural mechanisms that create both cognitive control and conscious awareness. Every moment of your conscious existence leaves traces in the neural networks of your brain, creating an ever expanding library of experiences that shapes who you are and how you navigate the world. Memory represents far more than a passive storage system. It is an active, reconstructive process that continuously shapes perception, guides decision making, and provides the foundation for personal identity. From the fleeting working memory that holds a phone number just long enough to dial it, to the deeply ingrained motor memories that allow you to ride a bicycle decades after learning. Memory systems permeate every aspect of brain function and human experience. The formation of memories begins with the encoding process, where patterns of neural activity are transformed into lasting changes in synaptic connections. This transformation occurs through mechanisms of synaptic plasticity that were first summarized by Donald Hebb's prescient observation that cells that fire together wire together. When neurons are active simultaneously, the connections between them strengthen, creating neural pathways that can be reactivated to recreate aspects of the original experience. The brain employs multiple memory systems, each specialized for different types of information and serving distinct functions in behavior and cognition. Declarative memory, also called explicit memory, encompasses conscious recollections that can be verbally described. This system includes episodic memory for specific personal experiences and semantic memory for general knowledge about facts and concepts. Non-declarative or implicit memory operates below the threshold of consciousness and includes procedural memory for skills and habits, as well as various forms of conditioning and priming. Episodic memory creates a personal timeline of experiences, allowing you to mentally travel back in time to specific moments and events. This remarkable capacity enables you to remember not just what happened, but when and where it occurred, along with the contextual details that made each experience unique. The hippocampus plays a crucial role in episodic memory formation by binding together disparate elements of experience into coherent memory traces that can be retrieved as unified episodes. The hippocampal memory system exhibits specialized neural mechanisms for encoding and retrieving episodic memories. Place cells in the hippocampus fire when animals are in specific locations, creating cognitive maps of spatial environments. Grid cells in the entorhinal cortex provide metric information about distances and directions, while border cells and head direction cells contribute additional spatial reference frames. Together, these cells create a sophisticated spatial navigation system that may provide the organizational framework for episodic memory. Semantic memory encompasses general knowledge about the world that has become detached from specific learning episodes. Unlike episodic memories, semantic memories lack contextual details about when and where they were acquired. The transformation from episodic to semantic memory may occur through repeated exposure and consolidation processes that extract general principles while discarding episodic details. The temporal lobe cortex, particularly the anterior temporal regions, plays crucial roles in semantic memory organization. These areas contain neurons that respond to specific categories of objects, concepts, or meanings, creating distributed representations of semantic knowledge. The organization of semantic memory appears to follow both categorical and featural principles, with related concepts clustered together based on shared properties and associative relationships. Procedural memory enables the learning and retention of motor skills, habits, and other behaviors that operate largely below conscious awareness. This memory system allows you to perform complex actions like playing a musical instrument or driving a car with minimal conscious effort once skills are well learned. The basal ganglia and cerebellum play central roles in procedural memory, with the basal ganglia contributing to habit formation and the cerebellum supporting motor learning and adaptation. The striatum, a key component of the basal ganglia, exhibits learning-related changes that support habit formation. During initial skill learning, the dorsal striatum shows widespread activation, but as skills become automatic, activation becomes more focused and occurs primarily at the beginning and end of behavioral sequences. This pattern suggests that the striatum learns to chunk behavioral sequences into efficient routines that can be executed with minimal cognitive control. The capacity limitations of working memory constrain the amount of information that can be actively maintained and manipulated, and then, most individuals can maintain approximately 7 plus or minus 2 items in working memory, though this capacity varies with the complexity of the information and individual differences in cognitive abilities. These limitations shape how complex cognitive tasks must be organized and performed. Synaptic plasticity provides the cellular foundation for all forms of learning and memory. Long-term potentiation, the persistent strengthening of synapses following repeated activation, represents the most studied form of synaptic plasticity. LTP involves both early phases that depend on post-translational modifications of existing proteins and late phases that require new protein synthesis and structural changes at synapses. The induction of LTP requires coincident presynaptic and postsynaptic activity, consistent with Hebb's rule for associative learning. NMDA receptors serve as coincidence detectors that respond only when both presynaptic glutamate release and postsynaptic depolarization occur simultaneously. This requirement for temporal coincidence allows synapses to strengthen specifically when pre- and postsynaptic neurons are active together. Long-term depression represents the opposite form of synaptic plasticity, involving the persistent weakening of synaptic connections. Limited may serve important functions in memory formation by eliminating irrelevant connections and preventing synaptic saturation. The balance between LTP and limited allows neural networks to maintain optimal levels of connectivity while preserving the capacity for new learning. Memory consolidation involves the gradual transfer of memories from hippocampal dependent storage to neocortical networks that can maintain memories independently. This process may take weeks to years for complex memories and involves repeated reactivation of memory traces during sleep and quiet waking states. Systems consolidation allows the hippocampus to continue forming new memories, while previously formed memories become integrated into long term cortical storage. Sleep plays crucial roles in memory consolidation through specific patterns of neural activity that occur during different sleep stages. Slow wave sleep features sharp wave ripples in the hippocampus that coordinate with cortical slow oscillations and sleep spindles generated by thalamic circuits. These coordinated oscillations may facilitate the transfer of information from hippocampus to cortex and the integration of new memories with existing knowledge. REM sleep may serve different consolidation functions, potentially supporting the integration of emotional memories and the extraction of general principles from specific experiences. The high levels of acetylcholine during REM sleep may promote synaptic plasticity and the reorganization of memory networks. Dream content often incorporates recent experiences in novel combinations, suggesting that REM sleep may support creative recombination of memory elements. Memory retrieval involves the reactivation of neural patterns that were active during encoding, but this process is constructive rather than simply reproductive. Each act of remembering can modify memories through reconsolidation processes that make retrieved memories labile and subject to updating. This malleability allows memories to be updated with new information, but also makes them vulnerable to distortion. The reconstructive nature of memory helps explain why eyewitness testimony can be unreliable and why personal memories can change over time. Schemas, generalized knowledge structures, influence both encoding and retrieval by providing frameworks for interpreting and organizing experiences. These schemas can lead to systematic biases in memory, with recalled events conforming more closely to schematic expectations than to original experiences. False memories can arise when retrieval processes generate vivid recollections of events that never occurred. These false memories can be as detailed and compelling as true memories, demonstrating that subjective confidence is not a reliable indicator of memory accuracy. Understanding false memory formation has important implications for eyewitness testimony, recovered memory therapy, and other situations where memory accuracy is crucial. Individual differences in memory abilities reflect variations in brain structure, function, and experience. Some individuals possess exceptional memory abilities, such as people with highly superior autobiographical memory who can remember detailed information about virtually every day of their lives. Other individuals, such as those with developmental amnesia, show selective memory impairments despite normal intelligence. Memory disorders provide insights into the organization and function of memory systems. Alzheimer's disease initially affects episodic memory while sparing procedural memory, reflecting the differential vulnerability of hippocampal and basal ganglia systems. Korsakoff syndrome produces severe anterograde amnesia along with confabulation, illustrating the importance of the mammillary bodies and related structures for memory function. Amnesia following hippocampal damage demonstrates the selective importance of this structure for declarative memory formation. Patients like HM, who underwent bilateral hippocampal removal to treat epilepsy, show profound anterograde amnesia for new episodes and facts while retaining the ability to learn new skills. These cases reveal the independence of declarative and procedural memory systems. The emotional modulation of memory involves interactions between memory systems and limbic structures that process emotional significance. The amygdala enhances memory consolidation for emotionally arousing events through connections with hippocampal and cortical memory systems. This enhancement helps ensure that emotionally significant events are well remembered, but it can also contribute to the intrusive memories characteristic of post traumatic stress disorder. Memory formation and retrieval involve widespread neural networks that extend far beyond traditional memory centers. Sensory cortices contribute to the storage of perceptual details. Motor areas store action related information, and prefrontal regions maintain strategic and contextual aspects of memories. This distributed storage allows memories to preserve the rich, multimodal character of original experiences. As we prepare to explore the brain's capacity for change and adaptation throughout life, memory serves as a foundation for understanding neuroplasticity more broadly. The mechanisms that allow experiences to create lasting changes in neural networks also enable the brain to adapt to injury, environmental changes, and new learning challenges throughout the lifespan. Memory formation represents just one manifestation of the brain's remarkable capacity for experience dependent plasticity. The human brain represents one of nature's most remarkable examples of adaptive architecture, capable of profound change and reorganization throughout the entire lifespan. Far from being a static computer programmed at birth, your brain continuously reshapes itself in response to experience, injury, and changing demands. This capacity for change, known as neuroplasticity, enables the transformation of a few thousand embryonic neural precursors into the 86 billion neurons of the adult brain, allows recovery from injury that would permanently disable a machine, and permits the acquisition of new skills and knowledge even in advanced age. The journey from embryonic neural development to mature brain function unfolds through a precisely orchestrated sequence of events that span decades. This extended developmental process distinguishes humans from most other species and underlies our exceptional capacity for learning and adaptation. Understanding brain development reveals not only how we become who we are, but also how the brain maintains its capacity for change throughout life. Neural development begins remarkably early in embryonic life, with the formation of the neural plate just 3 weeks after conception. This flat sheet of specialized cells folds to form the neural tube, which will give rise to the entire central nervous system. Along the length of this tube, different regions develop into the spinal cord, brainstem, and various brain structures, following genetic programs that have been conserved across millions of years of evolution. Neurogenesis, the birth of new neurons, follows different timelines in different brain regions. Most neurons destined for the cerebral cortex are born between the second and fifth months of human gestation, while neurons in other regions may continue to be generated into the postnatal period. The hippocampus may retain some capacity for neurogenesis throughout life, though the extent and functional significance of adult neurogenesis in humans remains debated. Once born, neurons must migrate from their birthplace in the ventricular zones to their final destinations in the developing brain. This migration can span enormous distances relative to cell size, with some cortical neurons traveling several centimeters to reach their target locations. Migration occurs along two main pathways: radial migration, where neurons follow glial fibers from the ventricular zone to the cortical surface, and tangential migration, where neurons move parallel to the ventricular surface to reach specific cortical areas. The cortical layering pattern emerges through an inside-out gradient of neuronal migration, with later-born neurons migrating past earlier-born neurons to form more superficial layers. This process creates the six-layered structure of the mature cortex, with each layer containing neurons born at specific developmental times. Disruptions in neuronal migration can cause serious developmental disorders, including lissencephaly and heterotopias that alter normal brain structure and function. After reaching their destinations, neurons begin the complex process of forming synaptic connections with appropriate target cells. Growth cones at the tips of developing axons navigate through the developing brain using molecular guidance cues that attract them toward correct targets and repel them from inappropriate ones. These guidance molecules include netrins, slits, ephrins, and semaphorins that create molecular gradients and boundaries throughout the developing nervous system. The formation of initial synaptic connections occurs through a combination of genetic programming and activity dependent processes. Neurons that fire together during development tend to strengthen their connections, while unused synapses are eliminated through competitive processes. This activity dependent refinement allows the developing brain to fine tune its connectivity based on patterns of neural activity that reflect both genetic programs and environmental inputs. Critical periods represent windows of enhanced plasticity during which specific experiences are essential for normal development. The visual system provides the best understood example of critical period plasticity. During the first few years of life, normal binocular vision is essential for the development of precise eye alignment and stereoscopic depth perception. Children who experience visual deprivation during this period, such as from cataracts or misaligned eyes, may develop permanent visual deficits even if the underlying problems are later corrected. The molecular mechanisms underlying critical periods involve the maturation of inhibitory circuits and the formation of paraneuronal nets that stabilize synaptic connections. During critical periods, high levels of brain-derived neurotrophic factor and low levels of inhibition create conditions favorable for synaptic plasticity. As critical periods close, increasing inhibition and the formation of extracellular matrix components reduce plasticity and stabilize existing connections. The prolonged development of the human brain creates extended periods of vulnerability, but also provides exceptional opportunities for experience-dependent plasticity. The prefrontal cortex, which supports executive functions and cognitive control, continues developing into the third decade of life. This protracted maturation allows extensive experience-dependent refinement, but also contributes to the increased risk-taking and emotional volatility often observed during adolescence. Synaptic pruning represents a crucial developmental process that eliminates excess connections to optimize neural efficiency. The human brain initially overproduces synapses, with peak synaptic density occurring during childhood before declining to adult levels through selective elimination. This pruning process is activity dependent, with frequently used synapses being strengthened and rarely used synapses being eliminated. The timing of synaptic pruning varies across brain regions, with sensory areas reaching adult levels of synaptic density earlier than association areas. The prefrontal cortex shows particularly protracted pruning that continues into early adulthood, potentially contributing to the gradual development of cognitive control abilities. Disruptions in normal pruning processes may contribute to various neurodevelopmental disorders. Myelination represents another prolonged developmental process that continues well into adulthood. The formation of myelin sheaths around axons increases conduction velocity and enables efficient long-range communication between brain regions. Different fiber pathways myelinate according to different schedules, with basic sensory and motor pathways myelinating early and association pathways myelinating much later. The corpus callosum, which connects the two cerebral hemispheres, shows particularly protracted myelination that continues into the fourth decade of life. This extended developmental timeline may contribute to the gradual improvement in interhemispheric communication and cognitive integration that characterizes human development. Similarly, long range association pathways within each hemisphere continue myelinating throughout adolescence and early adulthood. Human studies reveal similar effects of experience on brain structure and function. London taxi drivers, who must navigate the city's complex street layout, show enlarged posterior hippocampi compared to control subjects. Musicians who begin training early in life show structural and functional differences in motor, auditory, and somatosensory areas. These findings demonstrate that intensive training and practice can produce lasting changes in brain organization. The adult brain retains substantial capacity for plasticity, though the mechanisms and extent of plasticity change with age. Adult plasticity involves many of the same mechanisms as developmental plasticity, but operates within the constraints of existing neural architecture. Learning new skills, recovering from injury, and adapting to environmental changes all depend on adult plasticity mechanisms. Recovery from brain injury illustrates the remarkable adaptive capacity of the nervous system. Following stroke or traumatic brain injury, the brain can reorganize to compensate for damaged areas through several mechanisms. Intact areas adjacent to damaged regions may expand their function to take over lost abilities. Alternative neural pathways may be recruited to support impaired functions. In some cases, homologous areas in the opposite hemisphere may assume functions normally performed by damaged areas. Rehabilitation strategies increasingly target plasticity mechanisms to enhance recovery from brain injury. Constraint-induced movement therapy forces the use of impaired limbs while restraining unimpaired ones, promoting plasticity in motor areas, controlling the affected limbs. Intensive speech therapy following stroke can promote language recovery through plasticity in both damaged and intact language areas. Age-related changes in plasticity reflect both declining capacity for certain types of plasticity and the emergence of different plasticity mechanisms. While the rapid, experience-dependent plasticity of development decreases with age, adults retain substantial capacity for learning-dependent plasticity throughout life. Older adults may rely more heavily on bilateral brain activation and alternative neural strategies to maintain cognitive function. Cognitive training programs aim to enhance specific abilities through intensive practice, with mixed results regarding transfer to untrained tasks. While training can produce improvements in practice tasks, the extent to which these improvements generalize to other cognitive abilities remains debated. The most effective interventions may be those that target multiple cognitive domains simultaneously, or that provide training in naturalistic contexts. The molecular mechanisms underlying plasticity involve activity-dependent changes in gene expression that alter synaptic structure and function. Immediate early genes respond rapidly to neural activity and initiate cascades of gene expression that modify synaptic proteins. Late response genes produce structural proteins that enable lasting changes in synaptic connectivity and neuronal morphology. Epigenetic mechanisms provide additional layers of plasticity by modifying gene expression without changing DNA sequences. Environmental experiences can alter patterns of DNA methylation and histone modification that persist long after the initial experiences. These epigenetic changes may contribute to the lasting effects of early experiences on brain development and function. Sleep plays crucial roles in brain plasticity by promoting synaptic consolidation and homeostasis. During sleep, the brain undergoes systematic changes that strengthen important synapses while weakening unnecessary ones. The glymphatic system, which clears metabolic waste from the brain, is particularly active during sleep, potentially contributing to the restorative functions of sleep. Individual differences in plasticity reflect variations in genetic factors, environmental experiences, and baseline brain organization. Some individuals show exceptional capacity for plasticity, such as those who recover remarkable function following extensive brain injury. Others may be more vulnerable to environmental influences or show limited capacity for adaptation. Understanding these individual differences is crucial for developing personalized approaches to education, rehabilitation, and intervention. The changing brain challenges traditional views of neural development as a process that reaches completion in early adulthood. Instead, the brain emerges as a dynamic system that continuously adapts to experience throughout life. This perspective has profound implications for education, rehabilitation, and our understanding of human potential. As we turn to examine how sleep supports these plastic processes and maintains brain function, we carry with us an appreciation for the brain's remarkable capacity for lifelong change and adaptation. As darkness falls and your brain begins its transition towards sleep, a remarkable transformation occurs within the neural networks we have explored throughout our journey. The bustling activity of waking consciousness gives way to a different kind of neural symphony, one that serves not action and awareness, but restoration and consolidation. Sleep represents far more than simply the absence of wakefulness. It is an active, carefully orchestrated process that maintains brain health, consolidates memories, and prepares neural systems for the challenges of a new day. This nightly journey into unconsciousness reveals some of the brain's most essential functions, operating beyond awareness, yet vital for optimal cognitive performance and mental health. The cyclical nature of sleep and wakefulness reflects one of the most fundamental biological rhythms, driven by ancient molecular clocks that have evolved to anticipate the daily rotation of our planet. These circadian rhythms, from the Latin meaning about a day, coordinate not only sleep-wake cycles, but also fluctuations in body temperature, hormone release, and cellular metabolism throughout the 24-hour period. Understanding these rhythms reveals how deeply our brains are synchronized with the natural world, despite our modern tendency to live under artificial lights and irregular schedules. The master circadian clock resides within the suprachiasmatic nucleus of the hypothalamus, a small cluster of approximately 20,000 neurons that serves as the brain's timekeeper. These neurons exhibit intrinsic rhythmic activity with a period of approximately 24 hours, maintained by molecular feedback loops involving clock genes that regulate their own expression. The proteins produced by these genes accumulate during the day and are degraded at night, creating cellular oscillations that drive circadian rhythms throughout the body. Light serves as the primary Zeitgeber, or time cue, that synchronizes the internal clock with the external day-night cycle. Specialized retinal ganglion cells containing the photopigment melanopsin detect ambient light levels and send signals directly to the suprachiasmatic nucleus via the retinohypothalamic tract. This pathway allows the circadian clock to adjust its timing based on environmental light conditions, ensuring that internal rhythms remain aligned with the external world. The suprachiasmatic nucleus coordinates circadian rhythms throughout the body through neural and hormonal pathways. During darkness, signals from the suprachiasmatic nucleus trigger the pineal gland to secrete melatonin, a hormone that promotes sleepiness and helps maintain circadian timing. During daylight hours, melatonin secretion is suppressed, contributing to wakefulness and alertness. This melatonin rhythm serves as an internal signal of time of day that helps coordinate sleep wake cycles with other circadian processes. The transition from wakefulness to sleep involves coordinated changes in multiple brain systems that gradually shift neural activity from patterns supporting consciousness to those supporting sleep. The reticular activating system, which maintains wakefulness during the day, begins to reduce its arousing influence on cortical areas. Simultaneously, sleep promoting neurons in the ventrolateral preoptic nucleus become increasingly active, releasing inhibitory neurotransmitters that suppress wake promoting systems. Sleep itself unfolds through a series of distinct stages, each characterized by unique patterns of brain activity and serving different physiological functions. Sleep staging is typically based on electroencephalographic recordings that reveal characteristic brainwave patterns, along with eye movement and muscle activity measurements. These objective measures reveal that sleep is not a uniform state, but rather a dynamic process involving systematic changes in neural activity. Non-rapid eye movement sleep comprises the majority of sleep time and divides into three stages of increasing depth. Stage 1, the transition from wake to sleep, features a shift from the alpha waves of relaxed wakefulness to the mixed frequency, low amplitude theta waves, characteristic of drowsiness. This light sleep stage typically lasts only a few minutes and represents the gateway between consciousness and deeper sleep states. Stage 2 sleep features the emergence of sleep spindles and K complexes, distinctive waveforms generated by thalamic circuits. Sleep spindles are brief bursts of 12-14 Hz oscillations that reflect rhythmic firing in thalamocortical loops. These spindles may serve protective functions by blocking external sensory input from reaching consciousness, helping maintain sleep despite environmental disturbances. K complexes are large, slow waves that may represent responses to sensory stimuli that occur during sleep. Stage 3, also known as slow wave sleep or deep sleep, is characterized by high amplitude, low frequency delta waves that reflect synchronized firing across large populations of cortical neurons. This synchronization occurs when thalamic neurons enter burst firing mode, generating rhythmic waves that entrain cortical activity. Slow wave sleep predominates during the first half of the night and is associated with the deepest levels of unconsciousness and the highest arousal thresholds. Rapid eye movement sleep represents a paradoxical state in which brain activity resembles that of wakefulness, while the body remains paralyzed except for respiratory and cardiac muscles. The eyes move rapidly beneath closed lids, giving this sleep stage its name, while the electroencephalogram shows fast, low amplitude activity similar to waking patterns. Most vivid dreaming occurs during REM sleep, though dreams can also occur during other sleep stages. The muscle atonia of REM sleep results from active inhibition of motor neurons by brainstem circuits that prevent the acting out of dream behaviors. This protective mechanism occasionally fails, resulting in REM sleep behavior disorder, where individuals physically enact their dreams. This condition provides insights into the normal function of REM sleep motor suppression and demonstrates the complex neural mechanisms that normally separate dream experiences from motor output. The alternation between NREM and REM sleep follows a predictable pattern throughout the night, with cycles lasting approximately 90 minutes in adults. Early in the night, slow wave sleep predominates, while REM sleep becomes more prominent toward morning. This temporal organization suggests that different sleep stages serve complementary functions that are optimally timed relative to circadian processes. The glymphatic system represents a recently discovered brain maintenance mechanism that operates primarily during sleep. This system uses cerebrospinal fluid flow along blood vessels to clear metabolic waste products from brain tissue, including amyloid beta and tau proteins that accumulate in Alzheimer's disease. The glymphatic system is most active during sleep when brain cells shrink, creating larger extracellular spaces that facilitate fluid flow and waste clearance. Sleep deprivation studies reveal the critical importance of adequate sleep for cognitive function and health. Even modest sleep restriction can impair attention, working memory, and decision-making abilities. Chronic sleep deprivation is associated with increased risk of numerous health problems, including cardiovascular disease, diabetes, obesity, and neurodegenerative conditions. These effects underscore sleep's fundamental importance for maintaining both brain and body function. The neural mechanisms underlying sleep regulation involve complex interactions between circadian timing systems and homeostatic sleep drive. Adenosine, a metabolic byproduct that accumulates during wakefulness, serves as a chemical signal of sleep need. Caffeine blocks adenosine receptors, explaining its wake-promoting effects. During sleep, adenosine levels decline, reducing sleep pressure and preparing the brain for another period of wakefulness. Dreaming represents one of sleep's most fascinating phenomena, generating complex, often bizarre experiences during states of reduced external awareness. While dreams most commonly occur during REM sleep, they can happen during any sleep stage. Dream content often incorporates recent experiences, emotions, and memories in novel combinations that may reflect ongoing memory processing and emotional regulation. Narcolepsy represents a disorder of REM sleep regulation, where components of REM sleep intrude into wakefulness, causing sudden sleep attacks and cataplexy. This condition results from loss of hypocretin neurons in the hypothalamus that normally help maintain wakefulness. Understanding narcolepsy has provided insights into the neural mechanisms that normally maintain boundaries between sleep and wake states. The development of sleep patterns changes dramatically across the lifespan. Newborns spend much of their time sleeping, with REM sleep comprising up to 50% of total sleep time. Sleep patterns gradually mature throughout childhood, with slow-wave sleep reaching peak levels during adolescence before declining throughout adulthood. Older adults often experience earlier bedtimes, more fragmented sleep, and reduced slow-wave sleep, changes that may contribute to age-related cognitive decline. Circadian rhythm disorders can result from misalignment between internal biological clocks and external schedules. Jet lag occurs when rapid travel across time zones creates temporary misalignment between internal rhythms and local time. Shift work disorder affects individuals who must work during times when their circadian clock promotes sleep. These conditions demonstrate the importance of maintaining alignment between internal rhythms and behavioral schedules. Sleep's role in emotional regulation involves the processing and integration of emotional experiences during sleep. REM sleep may be particularly important for emotional memory consolidation and the regulation of emotional responses. Dreams may provide a safe environment for processing threatening or emotionally challenging experiences, contributing to emotional resilience and adaptation. Individual differences in sleep needs and preferences reflect variations in circadian biology and sleep homeostasis. Some individuals are natural larks who prefer early bedtimes and wake times, while others are owls who function better with later schedules. These chronotype differences have genetic components and may reflect adaptations to different environmental niches throughout human evolution. As we prepare to explore how the brain changes with age, sleep provides a crucial context for understanding brain maintenance and health throughout the lifespan. The restorative processes that occur during sleep become increasingly important as the brain ages and faces greater challenges from accumulated damage and reduced repair capacity. Sleep's role in clearing metabolic waste and consolidating memories makes it essential for maintaining cognitive function as we age. As we reach the final chapter of our journey through the neural landscape, we encounter perhaps the most poignant aspect of brain function, how this remarkable organ changes across the decades of human life. The aging brain presents a complex paradox, simultaneously accumulating the wisdom of experience while facing the inevitable challenges of biological decline. Understanding these changes reveals both the vulnerabilities and the resilience of neural systems, offering insights into how we might maintain cognitive health while accepting the natural progression of time's effects on our most precious organ. The aging process affects every aspect of brain structure and function, though these changes unfold gradually and unevenly across different neural systems. Some changes begin surprisingly early, with brain volume starting to decline in the third decade of life at a rate of approximately 0.2% per year. This atrophy accelerates with advancing age, reaching rates of 0.5% annually after age 60. However, these structural changes do not translate directly into functional impairment, revealing the brain's remarkable capacity for compensation and adaptation. Gray matter changes during aging reflect the complex interplay between neural loss, synaptic reorganization, and compensatory mechanisms. Cortical thinning occurs throughout the brain but affects some regions more than others. The prefrontal cortex, which supports executive functions, shows particularly pronounced age related changes, while primary sensory and motor areas remain relatively preserved. This differential pattern of aging may explain why complex cognitive tasks requiring executive control show greater age related decline than basic sensory or motor functions. The development of white matter hyperintensities, visible as bright spots on brain imaging, reflects small vessel disease and tissue damage that becomes increasingly common with age. While some white matter hyperintensities may be benign, extensive lesions can contribute to cognitive impairment and increased risk of stroke. The distribution and extent of these lesions vary considerably among individuals, suggesting that vascular health plays an important role in brain aging. Synaptic changes during aging involve both loss and reorganization of connections between neurons. While the total number of synapses declines with age, this loss is not uniform across brain regions or synapse types. Some areas maintain stable synaptic density throughout life, while others show significant reductions. The brain may compensate for synaptic loss through sprouting of new connections and increased efficiency of remaining synapses. Neurotransmitter systems undergo specific age related changes that affect different aspects of cognitive function. The dopamine system, crucial for motivation and executive function, shows marked decline with age, with dopamine levels decreasing by approximately 10% per decade after age 40. This decline may contribute to age related changes in reward processing, motor function, and cognitive flexibility. The cholinergic system, important for attention and memory, also shows age related vulnerability. Reduced acetylcholine levels and receptor density may contribute to attention deficits and memory problems in older adults. However, the extent of cholinergic decline varies considerably among individuals, with some maintaining relatively intact function well into advanced age. Neuroinflammation represents an increasingly recognized aspect of brain aging that may accelerate cognitive decline. Microglia, the brain's immune cells, become more reactive with age, producing inflammatory mediators that can damage neurons and synapses. This chronic low level inflammation may create a cycle where initial age related damage triggers inflammatory responses that cause additional damage. Despite these biological challenges, the aging brain demonstrates remarkable resilience through various compensatory mechanisms. Cognitive reserve, the brain's ability to maintain function despite pathological changes, varies considerably among individuals based on factors including education, occupational complexity, and lifelong learning. Individuals with greater cognitive reserve may maintain normal cognitive function despite significant brain pathology. The concept of successful cognitive aging recognizes that not all older adults experience significant cognitive decline. Some individuals maintain stable cognitive function well into their 80 seconds and 90 seconds, while others even show improvements in certain domains. These superagers provide insights into factors that promote healthy brain aging and cognitive resilience. The aging brain also shows evidence of reorganization that can enhance certain cognitive abilities. Crystallized intelligence, reflecting accumulated knowledge and skills, typically remains stable or even improves with age. Older adults often excel at tasks requiring wisdom, judgment, and the integration of complex information based on experience. This preserved function demonstrates that aging involves not just decline, but also continued development of certain capacities. Memory systems show differential aging trajectories that reflect their underlying neural substrates. Episodic memory, dependent on hippocampal function, typically shows age related decline, with older adults having particular difficulty remembering contextual details of experiences. Working memory capacity also declines with age, affecting the ability to manipulate information mentally and perform complex cognitive tasks. In contrast, semantic memory remains relatively preserved during normal aging, allowing older adults to maintain their vocabulary and general knowledge. Procedural memory for skills and habits also shows remarkable stability, explaining why older adults can maintain complex abilities like driving or playing musical instruments despite other cognitive changes. Attention changes during aging involve both declines and preserved abilities. Divided attention, requiring the simultaneous processing of multiple information sources, shows marked age related impairment. Older adults also experience greater difficulty filtering out irrelevant information and may be more susceptible to distraction. However, focused attention for simple tasks often remains intact, and older adults may actually show superior performance on tasks requiring sustained vigilance. Pathological aging involves neurodegenerative processes that exceed normal age related changes and lead to significant cognitive and functional impairment. Alzheimer's disease represents the most common form of dementia, characterized by the accumulation of amyloid plaques and tau tangles that damage neural networks crucial for memory and cognition. The progression of Alzheimer's disease follows predictable patterns, beginning in the hippocampus and spreading to cortical areas involved in memory, language, and executive function. Early symptoms typically involve episodic memory impairment, followed by difficulties with language, executive function, and eventually basic activities of daily living. Understanding this progression has led to staging systems that help track disease advancement. Other neurodegenerative conditions affect different neural systems and produce distinct patterns of cognitive decline. Parkinson's disease primarily affects dopamine producing neurons, leading to motor symptoms, but also cognitive changes affecting executive function and attention. Frontotemporal dementia involves degeneration of frontal and temporal cortex, producing changes in personality, behavior, and language. Vascular dementia results from cerebrovascular disease that damages brain tissue through strokes or chronic small vessel disease. This condition often produces a stepwise pattern of cognitive decline corresponding to vascular events, contrasting with the gradual progression typical of Alzheimer's disease. The distinction between normal and pathological aging remains challenging but crucial for clinical diagnosis and treatment planning. Mild cognitive impairment represents an intermediate state between normal aging and dementia, involving cognitive changes that exceed normal aging but do not yet interfere significantly with daily functioning. Some individuals with mild cognitive impairment progress to dementia, while others remain stable or even improve. Strategies for maintaining brain health during aging focus on modifiable risk factors that can influence cognitive trajectories. Physical exercise represents one of the most powerful interventions for brain health, promoting neurogenesis, vascular function, and cognitive performance. Regular aerobic exercise can improve memory, attention, and executive function while reducing risk of cognitive decline. Cognitive engagement through lifelong learning, reading, and mentally stimulating activities appears to build cognitive reserve and reduce dementia risk. Social engagement also plays important protective roles, with social isolation associated with accelerated cognitive decline and increased dementia risk. Maintaining strong social connections and community involvement may provide cognitive benefits through multiple mechanisms. Diet and nutrition influence brain aging through effects on vascular health, inflammation, and cellular metabolism. The Mediterranean diet, rich in fruits, vegetables, whole grains, and healthy fats, has been associated with reduced cognitive decline and lower dementia risk. Specific nutrients, including omega-3 fatty acids, antioxidants, and B vitamins, may provide particular benefits for brain health. Sleep quality becomes increasingly important for brain health during aging. Poor sleep can accelerate cognitive decline and increase dementia risk, while good sleep habits support memory consolidation and brain maintenance processes. Addressing sleep disorders and promoting healthy sleep habits represent important components of brain health strategies. Management of cardiovascular risk factors, including hypertension, diabetes, and high cholesterol, can significantly impact brain aging trajectories. The brain's high metabolic demands make it particularly vulnerable to vascular compromise, and maintaining good cardiovascular health supports optimal brain function throughout life. Emerging therapeutic approaches for age related cognitive decline include both pharmacological and non pharmacological interventions. While no medications can reverse normal aging, some drugs may slow cognitive decline in certain conditions. Cognitive training programs, brain stimulation techniques, and lifestyle interventions show promise for maintaining or improving cognitive function in older adults. The human brain stands as evolution's most remarkable creation, a 3 pound universe that gives rise to art, science, love, and the very thoughts that now process these words. From the molecular mechanisms that enable synaptic transmission to the vast networks that support consciousness, every level of brain organization reflects millions of years of evolutionary refinement. Understanding the brain enriches our appreciation for the complexity underlying every human experience. The simple act of reading this sentence involves the coordinated activity of visual areas that process letter shapes, language regions that extract meaning, memory systems that provide context, and attention networks that maintain focus. Behind every thought, emotion, and action lies the orchestrated activity of billions of neurons working in perfect harmony. This understanding also brings responsibility. As we learn more about how experiences shape brain development, how lifestyle factors influence cognitive aging, and how social and environmental conditions affect neural function, we gain both the opportunity and the obligation to create conditions that support optimal brain health throughout life. The aging brain ultimately teaches us about the continuity of human experience across the lifespan. While specific abilities may decline with age, the essential qualities that make us human, and remember, our capacity for love, creativity, and wisdom, can continue growing throughout life. The brain that once marveled at the world through a child's eyes can still find wonder in the accumulated understanding of age. As you now drift toward sleep, your brain will continue its essential work, consolidating the ideas encountered in this journey and preparing for tomorrow's challenges. The neural networks we have explored will orchestrate the restorative processes that maintain brain health, clear metabolic waste, and strengthen the synaptic connections that encode your experiences. In sleep and in waking, in youth and in age, the human brain remains our most precious possession and our greatest mystery. It is the source of our individuality and the foundation of our shared humanity, the vessel of memory and the architect of dreams. Through understanding its workings, we gain insight not only into ourselves, but into the remarkable nature of consciousness itself.